Ma'am, please start.
Yeah. The first two speakers are ready?
Yes, ma'am.
Yes, ma'am.
Yes. So, welcome all of you to the technical session 2 this morning. And we have a few talks, I think 5 talks in this session. And I thank the PMRF committee for inviting me to chair this session. Of course, let me introduce myself. I'm Professor Shravanti Chaudhary from the Department of Chemistry at ISL Pune. And the first talk will be given by Mahesh Rajashri from the Department of Computer Science and Engineering at IIT Kanpur. So it's on the cryptanalysis of, I don't know how you spell it, it's KCAC or I don't know.
KCAC.
Okay, KCAC and algorithms for lattice problems. So Mahesh, you have 15 minutes to deliver your talk. Over to you.
Thank you, ma'am. I believe my screen is visible.
Yes, yes.
Okay, hi everyone. I am Mahesh. I am a PMRF scholar from the Department of Computer Science and Engineering Department in IIT Kanpur. My supervisors are Professor Manindra Agarwal and Professor Shashank Mehta. So in this talk, I'll be presenting cryptanalysis of KCAC and algorithms for lattice problems. So in each section, I'll be giving a motivation, a brief introduction, and few results. So let's start with KCAC. So this is a snapshot of the Ubuntu download page. And in the middle, you can see a verify your download button. On clicking it, we can see this box, which says that if we run the command given in the gray box, it will verify whether the downloaded file is consistent with the original Ubuntu file. Essentially, what this command is doing is it's running this algorithm, SHA-256, on the downloaded file, produces a 256-bit string, and checks it with a reference string. This 256-bit string is called the hash of the file, and this algorithm, SHA-256, is a hashing algorithm. In fact, it is a cryptographic hash function, which is resistant to many attacks. So its practical applications include message integrity checks, digital signatures, authentication, etc. The SHA-256 algorithm is one of the algorithms in this SHA-3 family, and this is based on KCAC. So it is important that we study the security of KCAC to avoid any real-world attacks. Let me formally describe these attacks, a few of them. So let H be a cryptographic hash function. Then a pre-image attack is, given a hash value h of m for some message m, find a message m dash such that h of m dash is equal to h of m. That is, the hash of m dash is equal to the hash of the message m. A different attack is, which is kind of easier than the pre-image attack, is the collision attack. Here you just want to find two different messages, m dash and m, such that their hash values are equal. So let us look how KCAC works. So let m be your Ubuntu file. It is first padded by some string and then divided into blocks of r bits. The first r bits is XORed with 0 and given as input to the function f, along with another c 0 bits. So the input size of f is r plus c bits. Now the output of this function f is XORed with the second block of m, and it's given to a function f, and it continues like this until all the blocks of m are absorbed. Now the hash value is extracted by taking the last r bits of the last function f. Now, if you want more than r bits, you again run the f function and extract as many as bits you want. So let us look at this function f more carefully. So as I mentioned, the input to the function f is in C plus r bits. It's C plus r bits, and it is represented as this three-dimensional structure called a state. Now, the first function in f is a theta function. What it does is it updates a single cell by adding the neighboring 10 cells. Rho is the second function, and it simply shifts the entries in the state backwards by some offset. Similarly, pi just shifts it sideways and upwards. Chi is the only nonlinear function. Nonlinear in the sense that the entry is updated by taking there's an addition. Apart from that, there's a multiplication of two different entries. That's why it's called nonlinear. Observe that the previous three functions were just shifting or addition. And finally, we have the last function iota, which is just adding a fixed constant to the entry 0, 0. So essentially, these 5 functions are just mixing up the bits in the states. Now, the function f is nothing but the composition of these 5 functions, some r many times, where r is called the number of rounds. So in our work, we study nonlinear structures on Keswick. So what this actually does is it looks at this nonlinear function chi and try to find weaknesses in it. By doing so, we get a better pre-image attack, which is given in this table. So if you look at it, for 2 rounds, we have like an improvement 2 raised to 16 factor than the earlier non-result. Now let's look into the Zn isomorphism problem, which is actually a lattice problem. So most of the currently known crypto systems are based on RSA, Diffie-Hellman, and these are based on integer factoring or discrete log. These 2 problems are known to be hard for classical computers. But once you have a quantum computer, these can be easily solved using the Scholz algorithm. So these crypto systems can be easily broken when there's a crypto system, that is, when there's a quantum computer. And it is expected that in the near future, we can expect a quantum computer. So people have started to look into post-quantum crypto system, and lattices is one candidate. This is because we know hard problems like shortest vector problem, closest vector problem, which are hard for even quantum computers. So mathematically speaking, a lattice is defined by a basis B1 to Bn, which is a set of linearly independent vectors. And the lattice is nothing but the set of all integer linear combination of this basis vector. For example, let the basis vectors be 1, 0, 0, which is marked by this red arrow. Then the lattice spanned by these 2 vectors is this black dots, which actually extends to infinity also. There's a name for this lattice, it's called Z2. 2 because there are 2 entries, Z because it's just only integer components. So one thing that you can try to do is you can check whether these 2 vectors, that is 1, 1, 2, 1, also span the same lattice. And it's a pretty simple proof. So from this, we can come to know that a lattice can have multiple bases. In fact, it has infinitely many bases. Now, let us look at a hard lattice problem called the shortest vector problem. So in this problem, you're given a lattice, and you want to find a shortest non-zero vector in the lattice. Now, why non-zero? Because every lattice contains the 0 vector. That is evident from the definition of a lattice, because you can just set your integer linear combination to all zeros. So b times zeros is 0. So every lattice will contain the 0 vector. So you want to find the shortest vector, which is not equal to 0. Coming back to the Z2 lattice, we can see that these 4 vectors, that is 0, 1, 1, 0, 0, minus 1, minus 1, 0, are the shortest vector to the lattice. Now, why is SVP hard is because of the fact that a lattice has a lot of bases, infinitely many bases. So imagine I give a random lattice with a basis which has very long vectors. Then you don't have any information of the shortest vector in the lattice. And due to this fact, we only know exponential time algorithms for solving SVP. In other words, if you have like even small values for n, let's say like 500 or 1000, it will take millions of years to solve this problem. Now, the Zn isomorphism problem is you're given a lattice and you want to check whether this lattice is isomorphic to Zn. Mathematically speaking, you're given a basis B for some lattice, and you want to decide whether they exist an orthonormal matrix O and a unimodular matrix U such that O times B equal to U. Now, U is a unimodular matrix is another name given to a basis of Z. And an orthonormal matrix means it's kind of rotating the space. So you're asking whether you can rotate your lattice such that it will get aligned back to Z or something. For example, let us go back to the Z2 lattice. Now, if I rotate the Z2 lattice by 10 degrees, then I get this lattice, which is denoted by this red marks, red dots. So the Zn isomorphism is just asking whether I can rotate it back and get Z2. A point to be noted is that there are other lattices which kind of mimics Zn lattices. And even if you try to rotate it, it won't get aligned properly with the Zn lattice. Interesting facts about Zn isomorphism is that it is known to be an NP intersection co-NP. This simply means that it is not as hard as the SVP problem. In fact, we know an algorithm, a trivial algorithm, which can solve Zn isomorphism using SVP algorithm. But we have seen that SVP algorithm takes exponential time. So we want to see whether we can do something better than exponential. So to find better algorithms, we looked into the basis of Zn, and we gave the following theorem, which is if I start with a vector v whose norm is strictly greater than 1, then I can find b2 to bn vectors, which is again integer vectors itself, such that v b2 up to bn will form a basis of Zn. At the same time, these new vectors bi's are strictly smaller than the vector v. The proof of this theorem uses concepts from number theory, and one should know that this is only true for Zn lattice. And currently, we are working on an extension of this theorem, which states that if I start with v1 to vk integer vectors, such that everyone is strictly greater than 1, and without loss of generality, you can take that v1 is the largest. And these vectors can be extended to a basis, that is v1 to vn. It will form a basis of Zn. Then there exists bk plus 1 to bn, such that v1 to vk, that is the initial vectors, along with this bk plus 1 to bn will form a basis. And this bi's are strictly smaller than the largest vector v1. And with this, concluding my talk, I would like to thank all and would be happy to answer your questions. Thank you.
Thank you, Mahesh, for keeping very much on time. And any questions from any one of you? I have a small question.
Yes, ma'am.
When you first showed your algorithm, that pictorial representation, you told something about a pad. What is that you're talking about?
Okay, I'll share my screen once again.
In your first few slides.
Yes, I think over here, right? Yeah. So the padding, what happens is, I said that this message M with the padded version is divided into R bits. So the input file won't be a multiple of R bits. So to make it R, you add this random padding, which is 1 0 star 1. That is, I add 1 and enough number of zeros and then again a 1, so that the total size will be a multiple of R. Total size of M plus this padded string.
Okay, yeah, I can now see the connecting way you're showing in your pictorial representation. Yeah, thanks. Yeah. Any more questions? Okay, then let me thank Mahesh for for this wonderful talk, and keep going on with your good work. Thank you, Mahesh. And now you can, I'll call the next speaker, Rohit Chaudhary. Are you there, Rohit? Yeah, I can see you. Yeah. Rohit is from the Computational Data Science Department at IISc Bangalore. And today, Rohit will be talking about optimal planning of autonomous navigation vehicles in stochastic dynamic environments. Rohit, you have 15 minutes for your talk.
Sure. Just let me share my screen. Can you please confirm if you can see my screen?
Yes, we can see your screen.
Okay. Thank you. So, all right. So good afternoon, everyone. My name is, just one second. I have, yeah, right. So good afternoon, everyone. My name is Rohit Chaudhary. I'm from the Department of Computational and Data Sciences at IISc Bangalore. And today I'm going to talk about my work on optimal planning of autonomous vehicles in stochastic dynamic environments. Now, autonomous vehicles are in great demand because they're very useful in missions like data collection, surveillance and security operations, resource exploration missions, etc. And one thing that we require from them is that they behave optimally. And this brings me to the problem statement itself. It is an optimal planning problem. So let's consider an AUV, an autonomous underwater vehicle that must collect data by maneuvering around optimally in a stochastic dynamic oceanic environment, such as one shown over here. And it does so in order to estimate some field parameter. So let's say it wants to estimate the temperature field at a certain depth in this oceanic region. Of course, it's going to be limited by some constraints. It may have a limited mission time, a limited battery life. And so the first question we ask is that where and when in this environment should the data be collected? So that is asking to identify the points A and B. And then more importantly, we ask that how should this data be collected? As in, what is the optimal plan for the agent to traverse between an identified point A and an identified point B? And this is what we refer to as the A to B problem. And this problem is the focus of our current work. Now, to tackle this A to B problem, we use the Markov decision process framework, or the MDP framework, for the optimal control of these AUVs. Now, we use MDPs because they provide a natural framework for decision making in stochastic environments, but they can be computationally expensive. So, for example, this 2018 ICRA paper by Kularatne attempts to solve a similar A to B problem in this oceanic environment and reports a compute time of 13 hours on a CPU for a problem size of roughly 4 million states, which is a lot of time. So now we ask that, can we come up with computationally feasible solutions for the A to B problem? And we address this problem through two different approaches. So the first approach involves approximating the optimal policies or these optimal plans using reinforcement learning ideas. In the second approach, we try to accelerate the dynamic programming pipeline itself using a GPU, and this method, we name it as GPU accelerated planning. So now the problem setup is as follows. We have an oceanic environment, which consists of a stochastic dynamic velocity field V. It may also have a stochastic dynamic scalar field, like the radiation field from the sun incident upon this area. There are going to be dynamic obstacles as well. And in this problem setup, the agent must travel from some point A to some point B in an optimal fashion, following an optimal policy. So the first approach or the first work that deals with this problem is what we call physics-driven model-based Q-learning. Now, this work consists of the following modeling framework. We have three key components. Firstly, we have a data-driven probabilistic environment modeling technique, which helps us to compute n different realizations of the stochastic dynamic velocity field. So one such realization is shown over here in this double gyre flow scenario through these arrows. So this is the velocity field. Now, for each of those realizations of the stochastic velocity field, what we do is we use the Hamilton-Jacobi stochastic PDEs in order to compute exact time optimal paths. And if we do so for all the n realizations of the stochastic velocity field, we end up having a distribution of exact time optimal paths as shown over here. And finally, the key component is to use a reinforcement learning algorithm like Q-learning to learn an optimal policy from the experience that can be extracted out of this distribution of trajectories. So the overall algorithm now has two different phases. Phase one is an offline phase, which we call transfer learning, wherein we first compute this distribution of time optimal trajectories from the Hamilton-Jacobi equations. Now, for each of these trajectories, we figure out or we compute a sequence of states and corresponding actions that the agent must take so that it keeps following that trajectory. Now, if we do that for one trajectory and for all such trajectories in the entire distribution, we end up having with ourselves a experience buffer. Now, with this experience buffer, we can update the Q values using the Q learning algorithm and computing a good initial policy pi1. Next, we have the second phase, wherein we dynamically update the optimal policy in mission. So initially, the agent is going to start out executing the policy pi1 it had learned in the offline phase. Then once it goes out into the mission and collects some environment data, it's going to compute a posterior estimate of the velocity field. With this posterior estimate, it's going to perform future rollout simulations using an epsilon greedy Q-learning algorithm, which is to say that it explores some new action sequences to see that if it can do any better in this new posterior estimate of the velocity field. Then the next action is performed based on the updated Q values. So this keeps on going in loop, and as this loop continues, the agent keeps moving towards the target location while updating its optimal policy. So let's see how our algorithm compares to traditional dynamic programming approaches. So if we have a look at the distribution of trajectories that was obtained by executing the optimal policies computed in each case, we see that the distributions look very similar. Then if we have a look at the expected mission travel time for the following problem parameters, we see that the mission travel time by executing our policy, our optimal policy, is roughly 13.1 days, compared to the 12.6 days it would take if it followed the optimal policy from dynamic programming, which is once again very similar. But more importantly, if we have a look at the compute time, we see that our algorithm just takes around 11 minutes to compute the optimal policy compared to the 90 minutes it takes using traditional dynamic programming approaches, which really highlights the key, like the significant computational advantage that our work has achieved. So all of this was one method of solving the A to B problem in a computationally efficient manner. Now, let me move on to the second method, which is GPU accelerated planning. Over here, once again, we're solving the A to B problem, but we also have an additional stochastic dynamic scalar field as well. So this could be the radiation from the sun incident upon this oceanic region. And over here, the key idea is to accelerate the dynamic programming pipeline using a GPU and achieve speedups. But then why use a GPU in the first place? So firstly, for our problem, the transition probability matrices are sparse. And so we can use efficient libraries like Thrust and QSparse, which have very good parallel implementations of sparse matrix operations. More importantly, the phase of the algorithm where we build the model, that is where we compute the probabilities from the velocity field data in the first place, it is an embarrassingly parallel process, meaning that it involves a lot of independent computations. And so it's very suitable to use a GPU in such a case. And finally, we have the sparse value iteration. We use a modified version of the sparse value iteration algorithm due to Sapio et al., which is essentially a GPU implementation of the value iteration algorithm, but optimized for sparse matrices. However, the challenge over here is that GPUs come with limited compute and memory resources. So in order to overcome these challenges, the following solution ideas were implemented. So firstly, we use a reduced order velocity field, which helps us save a lot of memory. We prove mathematically that only the mean of the scalar field is required, which once again helps us save a lot of memory. Then we implement this idea of a neighboring subgrid, which essentially stems from the fact that an agent can only transition to spatially nearby locations and not anywhere arbitrarily in the domain. And finally, a lot of work went into developing efficient CUDA kernels. But I cannot go into these details right now because of lack of time. But suffice it to say that because of these solution approaches, what we were able to achieve are speedups ranging from 600 times to 1000 times for large problem sizes, depending on the kind of GPU we are using. So in terms of compute time, so let's say for a problem of roughly 8 million states, we see that our GPU accelerated planner could compute the optimal policy in roughly around 7 minutes. Which is really peanuts compared to the order of hours, multiple hours that it takes using sequential conventional methods. So there are multiple applications for our planner, such as single objective planning and multi-objective planning. So let me give you a quick example demonstration of a single objective problem, wherein we're trying to optimize the net energy consumption of the AUV in this oceanic environment. By net energy, I mean the consumed energy because of maneuvering around minus the collected energy. And the agent can basically collect energy if it has a solar harvesting device by moving around in sunny regions. So over here, the streamlines are the velocity field. The yellow region is the sunny region. The red region can be thought of as a cloudy region. And the agent must move from point A to point B optimally while avoiding these obstacles. So now what you see over here is a distribution of trajectories that was obtained by following the optimal policy computed with our planner. And we see that the agent initially moves towards the left, tries to remain in the sunny region as much as possible, trying to collect energy. And then once it's no longer feasible to stay in the sunny region, it starts moving quickly towards the target location while avoiding these square obstacles. Another interesting application is multi-objective planning, where we try to optimize multiple objectives. For example, the expected mission time and the expected energy consumption, which are sort of conflicting in nature. And what we want to do over here is to compute optimal operating curves, such as one shown over here, which shows us the trade-off between these two conflicting objectives. Now, in order to compute such a curve, we need to solve multiple such MDPs. And the significant advantage that our planner provides is that we can now compute multiple such operating curves in a matter of hours, compared to a matter of days, which would take if we use conventional sequential methods. And so the advantage is that with our planner, we can use the latest velocity field information to do the planning, which is really good. So in conclusion, we have developed some efficient methods of computing optimal policies for autonomous vehicles in stochastic dynamic environments. Our first method was a physics-based Q-learning algorithm, which computes a near-time optimal policy, achieving around 8 times speed up compared to dynamic programming methods, and has been published at the DVDS 2020 conference. Then we have the second method, where we use our GPU-accelerated planner to compute optimal policies and achieve speed ups ranging from 600 to 1,000 times. And we have 2 works under submission right now, under review. As far as future work is concerned, we're trying to implement some onboard routing techniques, which involves replanning and recomputing these optimal policies in mission. We're looking at efficient RL methods as well for tackling continuous state and continuous action spaces. And we're also looking forward to solve some pursuit evasion problems in stochastic dynamic environments. This brings me to the end of this presentation. Thank you so much for your time. I'd be happy to answer any questions.
Thank you, Rohit, for keeping up to the time. Any questions from anyone of the audience? Anyone in the audience? Okay, Rohit, I just had a quick question.
Yes.
That in this stochastic model that you're considering, you're only taking a Markovian description, correct?
Yes.
Yeah, this is a Markov state model, right? So so when you talk about this autonomous navigation vehicles in Vegas, so is there no is a Markovian description enough, or memory effects may come into play out here to can we totally rule out the non-Markovian character of this problem?
So let me share my screen again. So in our case, the Markovian description is enough because the state in our case is the position and time of the state is like X, Y, and T. So that is, so if you see this figure over here, so we have X axis, the Y axis, and the time axis. And also an assumption in this is that the state is fully observable. So it is sufficient to know where, in what time, and in what position our agent is to choose a future action or to choose a sequence of actions. So yes, for the way that we have set up the problem, a Markovian description is enough.
So you're not taking any memory effects in your system?
No, not
in
this.
Okay, thank you. Thank you. Now, thank you, Rohit. Let me move to the if there are no more questions, let us all thank Rohit, and I'll move to the next speaker, Adarsh Mukesh. Adarsh is from the Advanced Technology Development Center at IIT Kanpur. He'll be talking on deviant detection and spectral shape selectivity in the mammalian auditory cortex. Adarsh, you have 15 minutes to present. So over to you.
Good afternoon to everyone.
You are audible and we can see your screen. Okay?
Yes. I'll be presenting my work, deviant detection and spectral shift selectivity in mammalian auditory cortex. So the crux of my problem statement is to understand how the mammalian brain, it essentially detects rare events that are not that frequent in the auditory scene, and how complex sound features, how spectrally complex sound of objects, how are they encoded in the human brain. So a little background. So on the left, we see different functional components of the neural processing. So we see that from our senses, we pick up information from the environment, and then it goes up at through multiple hierarchical levels. And then it finally reaches the working memory part, wherein there is again a top-down control, that is higher order cortical regions of our brain are sending inputs to the lower order ones, and it acts as a feedback. So the two main control components here that we see, one is the top-down control, next is the bottom-up control. The next figure here, this is a rough diagram of the central auditory pathway. So it starts from the cochlea, that is just next to our external ear, and then it goes up till a part of the brain called as the auditory cortex. Sorry. Yeah. So in the cochlea itself, complex sounds are broken up into its individual frequency. We can say that a Fourier breakdown happens in the cochlea itself, and then this information is carried forward till the auditory cortex. One phenomenon I would like to introduce you to you is the cocktail party effect. So imagine that you are standing in a party and you are getting sounds from all different kinds of sources. Still, you are able to attend to one particular sound and are able to reject all the other. So the basic question is that what essentially decides this selectivity? Is the environment is in a way directing us to pick up only the relevant features, and if it's something like that, how is the relevant feature decided? Next, coming to somewhat of a literature survey. So there's a phenomena called as the SSA, stimulus specific adaptation, wherein we see that if one particular sound object is being repeated multiple number of times, which we call as a standard, then the response to that particular sound object, the standard, it decreases. And when something rare, which we call as a deviant, a rare event occurs, then the response for that it enhances. So what we did is that we performed similar experiments at very low age. Our model animal is mouse at very low age, that is P7, 7 days after the birth, wherein the animal, wherein the ear canal is completely closed. And what we found is that when we place such oddball paradigm, such standard deviant paradigm, we get responses, we get robust responses at very low age, as low as P7. So this implies that the architecture, the brain architecture that is essentially needed to detect these rare events is mature at a very lower ages. We also performed some confirmatory experiments where what we did is that we took very uh, juveniles from the age of P6 to P11, and we put them inside a box and we played and standard deviant kind of sound exposure. And after these five days, we kept the animal back in the controlled environment in the animal house, simple animal house conditions. And at later ages, what we found is that the synapses that were essentially carrying the information for the rare event, for the rare frequency, the deviant frequency, these synapses are strengthened. So essentially implying that this whole feature, this whole characteristic of picking up something rare from the environment, it's not something that is top down. It is something that is inherently present in the neural structure itself, and the machinery to get it, it matures at a very early age. So sum total of what we got from these experiments is that the detection of rare events, rare or auditory events, is a bottom-up phenomena rather than a top-down phenomena. And the auditory machinery is mature enough to detect rare events at a very early age. So the next challenge is that is it a simple way to explain this phenomena? Here comes a hypothesis called as the narrow frequency channel hypothesis. So I previously told that the complex sound is broken down into its respective frequencies in the cochlea itself, and then these information about the different frequencies, these are independently carried through the auditory cortex in narrow frequency channels that operate more or less independent of each other. Combining this theory with the theory of depressing synapse. So the concept of depressing synapse essentially says that the neurotransmitter present in each synapse, it is not unlimited, it is limited. And on successive activation of a particular synapse, what happens is that the neurotransmitter pool of that synapse, it starts depleting. So over here, you can see we have three different variables, XR, XE, XI. So these are the reserve pool, the effective pool, and the inactive pool. And the sum total of it is the total neurotransmitter pool. So what happens is that when we activate a particular synapse multiple times, the neurotransmitter pool doesn't get sufficient time to replenish itself. And that's why if a particular synapse is a particular frequency is repeated multiple times, the successive EPSCs, excitatory postsynaptic currents, subsequent EPSCs, they decrease in size. But when something rare happens, that is a new synapse is activated, because that synapse has not been depressed yet, that's why we get a very heightened response. So in this plot, we have simulated it computationally, and we have shown that using these two simple concepts, the narrow frequency channel hypothesis and the depressing synapse, we can essentially show SSA, tonal SSA, simple tonal SSA, where both standard and deviant are two separate tones. But there is a catch in this. Let's just say we perform a new set of experiments wherein we use broadband noise as standard and simple tone as deviant. So broadband noise is nothing but presence of all the frequencies with the same energy. So when we are presenting broadband noise multiple times, all the synapses are getting activated multiple times. So all the synapses should get depressed. So now if a tone comes, if a pure tone comes, we should not be getting any response at a deviant. But we do see such responses when a tone comes, and that too very heightened responses. Moreover, we also performed another set of experiment where we presented the same frequency, let's just say 12 kilohertz, but in standard it was louder, that is 20 dB, and at deviant it was a little softer, that is 5 dB. So the same synapse is getting activated multiple number of times, both at the standard and the deviant. Here, because the same channel is getting activated, we should not be getting any deviant response, but still we observe that we get heightened deviant response at heightened deviant even though the same channel is being activated. So essentially we come to the conclusion that the narrow frequency channel hypothesis, which is a widely accepted hypothesis, it is incomplete. It can only explain SSA, that is stimulus specific adaptation, deviant detection, when both standard and deviants are pure tone. So we must go back to the fundamental. Next, what we did is that we tried to see why should a neural circuit essentially attend to a rare event, what the neural circuit stands to gain. So I performed a theoretical derivation wherein I showed that if a neural circuit attends to the rare event mode, it increases the mutual information between the stimuli and the response. So essentially it is information from the basis of an information theoretic approach, it is by default, the neural circuit stands to gain a lot if it starts responding more to the deviant frequency. So this is something that we established, and this is irrespective of what kind of stimuli we are using, whether it is tone, whether it is noise. Next, we also wanted to see if, so till now we were seeing the stimuli as simple frequency like 12 kilohertz, 24 kilohertz. We wanted to see if there is another statistical metric, information statistical metric, that is more that can give us more information regarding the stimuli. Furthermore, there are another class of neurons called as inhibitory neurons, and we wanted to see if these inhibitory neurons essentially play an important role in encoding such complex statistical features. So the first set of experiments we did is that we tried to vary the temporal statistical feature of the oddball stimuli. So we played two different kinds of oddball stimuli. One is the periodic stimuli, wherein essentially we are getting 7 standard tones, that is either a tone or a noise occurring successively. On the 8th position, we are getting a deviant. So the deviant, the rare stimuli is occurring at the same position, and we are repeating blocks of this stimuli. So this we called as periodic. And another kind of stimuli, random, where the deviant, the rare event, is randomly occurring here and there. What we saw is that neural circuit as a whole in a network, it is able to encode such temporal statistical features by the fact that, we were getting different spike rates when we were playing these two different kinds of stimuli. Moreover, what we saw is that the inhibitory neurons, the PV neurons and the SOM neurons, they were essentially playing a very important role in this network level detection of periodic stimuli and the random stimuli. What we saw is that, so we also computed at a later stage, we also computed how the relative connectivity between the excitatory neurons and the inhibitory neurons, it rebalances. And what we found is that the PV neurons, the parvalvular neurons, which essentially project to the cell body, they show more hyperconnectivity when they are encoding regular stimuli, the periodic stimuli. But the other kind of inhibitory neuron, the SOM, it essentially showed hyperconnectivity when it is encoding random kind of stimuli. So we saw that both the inhibitory neurons were essentially showing some kind of network level rebalancing within themselves, which was essentially helping the entire network differentiate between the periodic stimuli and the random stimuli. So that's how we got to know that there are mechanisms through which complex temporal statistical features are they get encoded uh, inside inside the auditory cortex. Next, as we are talked that we are looking for some other metric which through which we can define the auditory stimuli, which it's, let's just say, a more relevant metric so that we can study the encoding of such features. So previous works exist where they have used auditory contrast. So contrast here essentially means the standard deviation of the energy levels of all the frequency, where people have used auditory contrast to study tuning properties. And here they show that the neurons in the auditory cortex, they essentially encode to specific contrast values, and they show specific selectivity to such contrast values. We performed similar experiments, but what we did is that we performed varying contrast stimuli in an oddball fashion, wherein there are obviously standard tokens and deviant tokens, but the standard here is essentially something that we called as the RSS, random spectral shape, wherein it consists of all the frequencies, but all the frequencies with varying contrast levels and varying energy levels. And we played two different kinds of stimuli, wherein the standard had more contrast and the deviant had less contrast, and the swap of it, that is the deviant had less contrast, standard had more contrast. Based on this stimuli, we got plethora of responses, and we categorized all these responses in 4 parts based on the context, that is what the neurons are preferring, at what stage they are showing more spike rate, at standard or deviant, and based on the preference of contrast, that is they are showing high spike rate for low contrast stimuli or the high contrast stimuli. So what we found is that the neurons that are showing low contrast preference at deviant, that is neurons that are preferring low contrast as rare elements, there the noise correlation, that is the relative connectivity between such neurons, is significantly less as compared to the neurons that are preferring high contrast. We also performed 2 photon imaging experiments where we essentially wanted to see what is the role of these inhibitory neurons, PV neurons and SOM neurons, in directing this specific contrast selectivity. What we found is that the neurons that were preferring low contrast and deviant, the neurons that were preferring low contrast as rare events, they were not at all getting any excitatory drive from the local connectivity, but they were receiving all the inhibition from the local connectivity. So it could be possible that the excitatory drive that these neurons are getting, it is coming from long range connections and not from the local connectivity. On the other hand, neurons that were preferring high contrast, they were the only ones that were getting excitatory drive from the local connectivity. Thus, sum total of all these experiments is that there is a preference for low contrast stimuli, but it could be possible that that preference is not being driven by the activity of by the local activity, but via long range connections. So these are in conclusion, I would like to say these are the, you can say, 2 major results that we got from our result. First, that there are essentially contrast channels, like there are frequency channels. It could be possible that there are parallel contrast channels also, which essentially do not take information of specific frequencies, but the contrast, or let's just say some higher order statistic that is present in the stimuli. And it could be possible that such encoding of such contrast activities, it is not at all dependent, it is less dependent on the local network activity and more dependent on long range connections. So these are the output. We have got 2 publications. The first one is, it essentially shows that detection of rare events, it is a bottom up process, wherein we showed that at very early ages, this machinery to detect rare events, it is mature and working. And we also showed that, and there is another publication where we showed that long scale adaptation, that is the detection of temporally varying statistical features in the stimuli. Inhibitory interneurons have a significant role to play on them, and there are separate subnetworks in the local network only, where which essentially help to detect such varying temporal statistical features from the stimuli. Thank you.
Okay, Adarsh, thank you. I think we have to you just you don't have any time much.
Oh,
So I will move to the next speaker. Um, Adhyara Sri Teja from the MBU department at ISC. Are you there?
Yeah, can you hear me?
Yeah, I can hear you. So we'll be talking about structure, function, and dynamics of bioactive peptides by NMR spectroscopy. So you have 15 minutes and please be on time.
Yeah.
Can you please share your slides?
So can you see my slides? Yeah.
You can start.
Good afternoon, everyone. I'm Sri Teja. I'm working in molecular biophysics unit under the guidance of Professor Siddharth P. Sharma. I've been working on determining the structure, function, and studying the dynamics of bioactive peptides using NMR spectroscopy. So there have been 3 peptides that I've been working on. The first one is urumin. So before getting into urumin, I would like to give a brief introduction on influenza virus, which is a negative sense single-stranded RNA virus, and is the schematic on the right of influenza A virus. The influenza viruses are divided into 4 types: A, B, C, and D. And IAVs, which is influenza A viruses, are known to cause the flu pandemics. As you can see in the schematic, the envelope is embedded with 3 proteins mainly: M2 ion channels, hemagglutinin, and neuraminidase. Till date, 18 subtypes of hemagglutinin and 11 subtypes of neuraminidase have been identified, and vaccines and antiviral drugs are the only existing ways to manage and treat influenza virus pandemics, which are also known as flu pandemics. But the problems with these existing ways are that there have been rising drug-resistant virus mutants, especially to the existing anti-influenza drugs that are M2 ion channel inhibitors and neuraminidase inhibitors. And the problem with vaccines is that we need to invest a lot of time in the research and development of a vaccine which is specific to this seasonal influenza A virus pandemics. So what are the alternatives that we can try is to target and attack the conserved domains of these proteins on the embedded in the envelope, which is particularly the stalk region of hemagglutinin. So in order to find the molecules, the best resource is host defense peptides. So host defense peptides are those peptides that are secreted and or made in order to hunt for food or in order to defend themselves by organisms. So, and urumin is one such host defense peptide which is secreted by Hydrophiliax bohovistara, an endemic frog species of Western Ghats, India. Our collaborators, Jacob Joshi and group, have shown that this 27 amino acid long peptide had shown virucidal activity against hemagglutinin 1 H1 subtype of IAV. It has shown inhibitory activity against all drug-resistant and drug-sensitive strains of H1N1. And they have also shown that it binds to the conserved stalk region of hemagglutinin 1 with very high specificity. So our objectives in this particular project is to express and purify isotope labeled urumin peptide in order to determine its structure using NMR spectroscopy, and then proceed to study the binding mechanism of urumin with hemagglutinin using NMR spectroscopy. So in order to achieve that, we went ahead and made a fusion protein construct containing a 6-histidine N-terminal tag, followed by a fusion protein cytochrome B tag, and followed by linker and our protein of interest, that is urumin. So the purification protocol that I'm showing here is the protocol that I followed for all the peptides I'm going to present today that I've been working on. So the plasmid of interest is transformed into BL21 E. coli cells, which have been grown at either 37 degrees or 20 degrees centigrade, depending on the peptide, followed by nickel affinity chromatography, and post cyanogen bromide cleavage, the cleaved the consonant peptides or the urumin have been purified using size distribution chromatography and reverse phase PLC, and finally analyzed and used using NMR spectroscopy. So this is the reverse phase HPLC chromatogram of urimine, both in its oxidized and reduced form. Urimine, as such, when expressed biosynthetically in Bl21D3 cells, is expressed in its oxidized form. That is, the two cysteines form a disulfide bond. And when added 1.1 millimolar TSEP, all the cysteines get reduced, and as a result, there is a change in the retention time on the reverse phase HPLC column. This has also been confirmed using mass spectrometry, MALDI-MS analysis, whereas the expected mass was 2961 Daltons, and the observed mass was 2958 Daltons. To further determine and to be assured that the cysteines are oxidized, we have done the reduction alkylation reactions using only a alkylating agent with the protein and also with both reducing and alkylating agent. From this result, we have concluded that the biosynthetically made urimine in the lab is indeed in its oxidized form. So we have acquired a 1D spectrum on the unlabeled urimine peptide and compared it with the synthetic urimine that has been made in using protein synthesis in chemically. And also when you compare the recombinant urimine that has been reduced with the synthetic urimine, they match pretty. Recombinant urimine, its oxidized form has very broad peaks. This slide broadening can be ascertained to the change in the dynamics of the peptide upon cyclization because of the disulfide bond. So we went ahead and made 59 labeled urimine peptide and acquired 59 HSQC. The spectrum shown in the blue on the right is the HSQC spectrum of urimine. You can see that there is a lot of peak overlap and line broadening observed in this. If you count the major intense peaks, that matches exactly with the expected number of peaks, that is 31. And the projection of this particular HSQC spectrum along the proton dimension, it matches well with the unlabeled unlabeled urimine peptide that has been made. So this showed that the major conformer is indeed the major conformer is present, but we do not know what this peak overlap or broadening is coming from. So assuming that it might be due to aggregation, we tried reducing the concentration and also reducing the temperature, and also added increasing concentrations of defomethanol. And even though all these conditions did not change much in the line broadening and peak overlap, it did improve the peak dispersion in the spectrum. So till now we are so I am at this point where I need to optimize the conditions in which the NMR data should be acquired in order to reduce the peak broadening and peak overlap. Once the optimum conditions have been obtained, I'd be proceeding to make the double labeled, that is N15C13 labeled sample in order to acquire triple resonance data and determine the structure. And the two other peptides that I'm working belong to the genus Conus, that is from marine cone snails. So these are rare cysteine framework cone snail peptides. So in order to give a brief introduction, why cone snail peptides? So the Conus peptides, which are present in the cone snail venom, are very rich in sequence and structural diversity, and also they have very high functional diversity, which is ascertained to this particular sequence and structural diversity. And the very rigid and high selectivity and stability of these peptides can be ascertained to the presence of unique connectivity in these disulfide-rich peptides. Often we have seen that many of the cone snail peptides have been used as drugs or drug leads or as structural scaffolds in drug design. So these cone snail peptides have been divided into 12 families based on their pharmacological targets, and also into 33 cysteine frameworks shown on the right based on their arrangement of cysteines in the peptide sequence. So why study these disulfide-rich peptides? Not only cone snail peptides, but many of the animal toxins such as scorpion toxins and spider toxins have been shown to have unique disulfide connectivities because of the cysteine-rich sequences. This disulfide bond framework is very crucial for their function and structural integrity. And the problem arising in increasing number of cysteines is the possible complexity of the possible conformers. Increasing complexity in the possible conformers. So shown on the right is the sequence of AR1446, which is a cone snail peptide, which contains 6 cysteines. So there are 15 possible combinations of disulfide connectivities, of which only one, shown in the blue box, is prevalent in this peptide in nature. So till date, there are only two techniques that have been used, that is either X-ray crystallography or mass spectrometry, in order to determine the disulfide connectivity of these peptides. But the problems with these two techniques is that in case of X-ray crystallography, you need a crystal of this peptide in order to determine the structure. And in mass spectrometry, there is huge amount of data that needs to be analyzed, and also it also depends on the how well a fragment or the peptide can be charged or can perform in the time of flight experiments used in MALDI-TOF. But there have been none of the existing NMR methods also give a variable insight into the disulfide network. So our objectives here lie in designing an NMR method which could give a correlation between the three bonded C-beta carbons across the disulfide bond of the cysteines. So in order to do that, we need peptides, and in order to get an estimate of what kind of J coupling values that we are seeing across the cysteine C-beta carbons in the cysteines across the disulfide, we have done theoretical calculations in order to determine the 3-bond J coupling constant between the C-beta and C-beta carbons using Gaussian 16, which has been done under the guidance of Dr. Jayashree. So this, the plot on the right shows the J values that have been obtained from the Gaussian 16 calculations. The CSSE dihedral angle has been swept from 35 to 135 degrees, and most prevalent in most of the peptides or in proteins is the angle that is around 90 degrees. So the minimum J value and the maximum J value within plus or minus 5 degrees around 90 is the value shown here, which is quite small, but we are still designing and optimizing the technique in order to get a correlation between these C-beta carbons. So in order to try and test this pulse sequence on proteins and how well it works, we have went ahead and got two sequences, with one with a known disulfide connective, one with unknown. The first one is Conus austini, which is CIU 25 from Conus austini, which belongs to framework 25. Sorry for the misalignment here. So this is the sequence of the consonant peptide, and the purification protocol has used to purify this particular peptide is the same as I mentioned before. And the peak in between the red dotted lines in the reverse phase HPLC chromatogram is the peak corresponding to CIU 25, and this is the corresponding mass spec for the particular peak that's shown on the left on the RP-HPLC chromatogram. This is the mass corresponding to the double labeled CIU 25 peptide. So using the double labeled sample, that is N15C13 labeled sample, I have went ahead and acquired the following experiments: HNCSEB, CBCSUNH, CCCUNH, HNCO and HNCSEO, which have been used to acquire to perform the sequential backbone assignments. So the strip plot on the right is a representative strip plot of the sequential, how the sequential backbone assignment has been done using HNCSEB and CBCSUNH, and the entire backbone of the peptide has been assigned using the following experiments that have been stated here. So the second peptide is CPT9, that is from Conus bertullinus, which belongs to framework 9. So the connectivity of framework 9 is known, which is 142536. So we went ahead and got the fusion construct and expressed the protein. The oxidation and reduction state of these peptides after expression have been tested and found to be in their oxidized form, that is all the 6 cysteines form a disulfide bond. So when purified on a reverse phase HPLC chromatographic column, C18 column, we found that it gives two distinct peaks, that is around 1.5 minutes apart on a 1 ml per minute elution, gradient elution, peak 3 named peak 3 and peak 4 here, which both of them gave us the same mass, that is very close to the that is the expected mass, 3232 Daltons. So we went ahead and acquired a 1D NMR spectrum of these two peptides, these two isoforms of the peptide, CBT9, and the peaks were a little broad in both the peptides. And also there have been distinct regions and differences in the 1D amide region of the both the isoforms. From this, it can be concluded that these two isoforms of the peptide do have some kind of differences in them in their conformation. So in order to conclude, the entire backbone of CIU peptide has been assigned. The CD spectrum, which I have shown here of both CAU25 and the isoforms of CBT9, indicated that they predominantly contain coil conformations. And the future directions of this is to use these express peptides in order to determine and test and optimize the pulse sequence that have been designed to determine the disulfide connectivities. Once it has been determined, it will be corroborated with partial detection and signalation followed by mass spectrometric analysis. And also, in order to I need to verify if the two peaks observed on the RPHPLC for the CBT9 are the two isoforms of the same disulfide connectivity or with two different connectivities, and further get double labeled sample and aqueous triple resonance data and determine the functional targets of both these peptides and study their mechanism of action. Thank you.
Thank you, Shudeja. So, any questions? Okay, if there are no questions, I think time is also up. We'll move to the, thank you once again. We will move to the next speaker, Anjali Bariyar. She'll be talking on replication, transcription, collisions, and intrinsic trigger of enzyme mutations. Anjali is from the School of Biology, ISET Trivandrum. Anjali, your time starts.
Thank you, ma'am. Could you please let me know if the cursor and screen is visible?
Yeah, it is visible.
Thank you, ma'am. So, good afternoon, everyone. Myself, Anjali Warrior. I'm a graduate student from School of Biology in ISET Thiruvananthapuram. First of all, I would like to thank the PMRF scheme, Ministry of Education, and the organization committee of the Symposium for the wonderful opportunity. Today, I'll be talking about the work we have done in which we investigated a molecular phenomenon called replication transcription collisions and how these events could lead to the generation of a very deleterious mutations called indels. I would like to begin with drawing your attention to the vast diversity of life around us. Ranging from the tiny bacterium to the mighty elephant, including us humans, all the living organisms are so diverse, yet there is one feature common to all of us, that is the unit of life or the building block of life, the cell. A cell can be imagined as a very tiny but efficient factory in which multiple machinery are operating to perform cellular processes. Listed here are a few of the important cellular processes constituting the central dogma of life. Replication is the process in which genetic material of our cell, the DNA, is duplicated so that information can be passed down to generations. Transcription is the process in which this information is converted into the form of RNA. Finally, this decoding process is completed by translation, wherein the information is translated to form proteins, the workforce of cell. As we can see here, very often, many of this machinery use the same template and occur at the same time, leading to the possibility of a physical encounter between these machinery. Let's think of our DNA as a very busy highway with heavy traffic, where transcription and replication can occur at the same time in the same orientation, leading to a collision in the co-directional fashion as shown here. On the other hand, transcription and replication can occur in the opposite orientations, leading to a more severe form of collision simply due to the relative impact called head-on collisions. These events were beautifully captured by electron microscopy in 1992, where this black arrow indicates the moving replisome, and awaiting the replisome is a heavy traffic jam of multiple RNA polymerases, converting this DNA into RNA. Both co-directional and head-on collisions are known to have negative consequences affecting both the machinery involved. And head-on collisions have a higher negative impact due to the relative impact between the machinery, as I mentioned earlier. So the replication fork can experience stress in the form of stalling or collapse of the fork, and many a times the transcription complex face premature termination, and the tract on which these machinery travel, that is the DNA, suffer breaks and other DNA damages. All of this finally affect the genome stability of the cell and result in mutations. What are mutations? Mutations are heritable changes in the sequence of DNA, and we'll further look into mutations by taking an example of the sentence with 3-letter words. This is much similar to the way in which that information on DNA is read in a triplet codon fashion or 3 nucleotide bases at a time. Any base change or any change in any of these bases in the sentence will result in the loss of meaning to an extent, such as shown here. These classes of mutations are called base substitutions. On the other hand, if the position of the base is affected by insertion of another base or deletion of this base, the meaning of the sentence is lost to a greater extent, and such kinds of or types of mutations are called insertion or deletion respectively, together indels. All the classes of mutations have both positive and negative consequences, like driving the force of evolution or causing deleterious diseases like genetic diseases or cancer. But how often do these events occur, or how often does genome mutate? Let's take the example of a simple bacterium with 10 million bases in them. Studies have estimated the rate of mutations to be 1 mutation in every 1,000 bases per replication round, and 10 billion such rounds can happen in the span of 12 hours. That results in 10 million mutations in 10 million bases. That means every single possible non-lethal mutation will occur in a matter of 12 hours. Coming into human context, spontaneous mutations have been studied to be a major leading cause of human cancer. As we can see here, although hereditary and environmental factors do exist, replicative errors contribute to driving the cancer most. That means a simple mistake in the machinery that we saw earlier can trigger oncogenesis. And analysis of the types of mutations seen in cancer studies have shown that a large proportion of them end up being indels. But we are yet to understand what intrinsic factor in the cell triggers the formation of such deleterious mutations. Here, our objective is to decipher the mechanism of generation of indels by collisions, which is an unavoidable phenomenon in any cell where replication and transcription are actively happening, and thus very well could be an intrinsic trigger for mutagenesis and oncogenesis. For this, we will be looking at factors from all the possible angles, like alternative DNA polymerase, which could be helping the stalled replisome, or transcription factors, which could be taking care of stalled RNA polymerases, or DNA repair factors, which could be taking care of the consequences of collisions. So to study mutations, we need to measure mutations. And for this, we have established a very simple but robust mutation reporter system in our lab. Simply, the presence of a reporter gene type E3 makes the cells sensitive to the antibiotic trimethoprim, and any loss of function mutation in the gene will inactivate it and thus make the cells resistant to trimethoprim. Using this reporter system, we carry out large-scale mutation assays called Luria-Delbruck fluctuation tests with wild type as well as deletion mutants. So wild type will have a complete set of genes, and deletion mutant will lack the gene of interest. So a comparison between them will let us know about the function of the gene. So we set up large-scale fluctuation tests with these strains and let them grow so that random spontaneous mutagenesis will occur. And these mutations are scored by selecting them on trimethoprim, plates containing trimethoprim. These are counted to measure the mutation rate, and they are collected and sequenced to know the mutation spectrum or the types of mutations. So the first factor that we looked at was an alternative DNA polymerase called Pol4. Pol4 is an error-prone polymerase and has a particular feature of being conserved from bacteria to humans across all the kingdoms, suggesting a very important function. First of all, we looked at the role of Pol4 in genome stability by subjecting wild type as well as Pol4 deletion strains to DNA damage through ultraviolet irradiation. We can clearly see that deletion of Pol4 makes the cells highly susceptible to DNA damage by UV, suggesting the role of Pol4 in genome stability. We went on to see the role of Pol4 in spontaneous mutagenesis due to collisions. So here we have measured the mutation rate of base substitutions and indels in both wild type as well as deletion of Pol4 strains indicated by the red bar. Here we can see that there is a 200% dramatic increase in the rate of indels, suggesting that alternative DNA polymerase do protect the genome from genomic instability, and specifically this is done by preventing the deleterious mutations called indels. This is more evident when you look at the spatial distribution of indels, where in the wild type case, you see a basal level of indels which we attribute to the presence of Pol4, which could switch with the major replicative polymerase and thus facilitate a smooth progression. But what happens when the cells lack Pol4 is that there is an accumulation of elevated levels of indels, and as indels as a proxy for collisions, we hypothesize that this could be because when Pol4 is not available for switching, the replisome is experiencing frequent and increased stalling, thus suggesting that Pol4 is indeed facilitating the replisome progression. But here we can see, even if Pol4 is switching with the major replicative polymerase, still the stalled RNA polymerase could be a roadblock for the progression. So how are these RNAP roadblocks removed? Here we are focusing on a transcription factor called MFT, which is known to bind to such stalled RNA polymerases and remove the same. Again, we looked at the importance of MFT for genome stability by treating them with a DNA damaging agent and anti-cancer drug called mitomycin C. Here, represented by the bold line wild type survival, we can see, and there is a drastic decrease in the survival of MFT deletion strains represented by the dotted lines. When we look at the context of spontaneous mutagenesis due to collisions, we can see that similar to Pol4, there is a 100% increase in the case of indels when the cells lack MFT. This suggested, the similar trend suggested that there could be a coordination between these two factors, wherein MFT could be removing the stalled RNA polymerases so that Pol4 can switch with the major replicative polymerase and ensure a smooth progression. This is exactly what we saw when we compared the mutation rate between wild type, single deletions of Pol4 and MFT, and the double deletion, wherein again we see that indels are significantly increased when the cells lack these factors, suggesting that Pol4 and MFT could be coordinating to alleviate the consequences of collisions by preventing indels. But what happens when the cells lack these factors? Such unresolved collisions are known to exacerbate genome instability and ultimately result in mutations like double-strand breaks. So we focused on a DNA repair factor called RECA, which is known to bind to such double-strand break regions. We wanted to know if RECA-mediated pathways are coming into play when cells have unresolved collisions due to lack of factors we identified earlier. For this, we measured indel frequency in wild type and Pol4 deletion strains and compared to that of RECA mutant and a double mutant with Pol4 and RECA absent. Here we can clearly see that the increase in indels that we saw in case of Pol4 deletion was drastically reduced when the cells lacked RECA, suggesting that RECA was the factor acting in the absence of Pol4 and leading to generation of indels. We verified the same using fluorescence microscopy, where we fused the RECA protein with green fluorescent protein, and each foci here represents binding of RECA molecules to double-strand breaks on DNA. The quantification on the right clearly shows that whenever the cells lack Pol4, there is an increased percentage of cells with RECA GFP foci, suggesting that RECA-mediated error-prone replication restart will act as a backup in the absence of Pol4. With this, I would like to conclude our model, wherein we know that the collision between replisome and RNA polymerase occur in cells, and the machinery could be stalled due to these events. We believe patrolling factors like Pol4 and MFT could act as patrolling officers by taking care of these damages. MFT will initiate the process by removing the stalled RNA polymerase, and Pol4 will restart the process of replication and thus ensure smooth progression or further progression of the replisome. When cells lack these factors, alternative repair factors like RECA will come into play and will repair the damage, but at the cost of generating very deleterious mutations like indels. With this, I would like to conclude our model in which we are suggesting a mechanism for anti-mutagenic resolution of replication transcription collisions, and also the identified factors, lack of identified factors will lead to an incoordination of replication and transcription, leading to mutagenesis and ultimately result in processes like oncogenesis. With this, I would like to thank the PMRF scheme, Ministry of Education, Organization Committee, Professor Srabanti once again. Also, my doctoral committee members, Professor Murthy Srinivasula, Dr. Nishan Keti, my mentor, Dr. Shabari, and all my lab members for the support. Thank you. I'll be happy to take any questions.
Thank you, Anjali.
Thank you.
for the nice presentation. Your slides were very good.
Thank you.
And yeah, I think, is there any questions? We are there to take up. We are there to take up one question. Okay, I think there are no questions. So Anjali, thank you once again for your presentation. I would like to thank all the speakers, all the 5 speakers in this session for their presentations and their wonderful work. And I'd like to thank the organizers for inviting me to chair this session once again. And over to the organizers.
Okay, thank you, ma'am.
Thank you. Yeah. Can I exit the meeting?
Yes, ma'am.
Yeah, thank you.
Hello.
Hello. Can you check your sharing?
Yes, I will just do that. Can I take a minute to just set up my screen and everything?
Okay.
Yeah, thank you.
Hello.
Hello, ma'am. Can we check your sharing?
Yes, I'll just. Can I share my screen once and just check if it's
Yes,
all right?
yes, ma'am.
Okay. Is my screen visible?
Yes, ma'am. Everything's fine, ma'am.
Okay. Thank you.
Please unmute yourself, sir.
Hi, should I just share my screen and test as well?
Yes, sir.
All right, just give me a second. Is it visible properly?
Yes, sir. Please change your text.
Yeah, just a second. All right.
Okay, that's fine, sir.
All right, yeah. Thank you so much.
Hello. Are the organizer somewhere around?
Good afternoon, sir.
Hello. Yeah, this is Professor Paul Raj.
Yes, sir.
Yeah. So, are the students ready? Are the presenter ready?
Sir, only two presenters there, sir. This session, first and third, sir. Meantime, sir.
Okay.
Dr. Darshan Shah and Dr. Banerjee.
Okay.
Oh, hi, Professor Paul Raj, Darshan here.
Yeah. Darshan. Very good. Afternoon.
Good afternoon, sir.
So, I don't know what about the other speakers. Hopefully they'll join.
So are we on schedule?
Yeah. The schedule is as
Yeah,
per.
start sharp 2:50.
Okay, great.
We'll start, right? The organizer will start, right?
1 minute, sir, please.
After 2:50 we'll start, right?
Yes, sir.
Should I share my screen and be ready, or
Wait, sir. Announce your name and then you share.
Okay, cool. Thank you.
So one speaker more join, Dr. Kantlesh Agrawal. Fourth one. Okay, sir. Can we start, sir?
Start, yeah. Very good afternoon, everyone, and welcome to this afternoon session of this PMRF Symposium 2022. And this afternoon technical session too, we have 5 speakers, and I think most of them are will be joining. A couple of them are here, and hopefully they will also join this thing. And every speaker is given 15 minutes time, and maybe they can finish it with 12 minutes or so. Then we have a more time to interaction. All right. With that, I think my name is Paul Raj. I am working as a professor in the School of Environmental Sciences. Basically, I'm a biologist and working in Jawaharlal Nehru University, New Delhi. So this is my introduction, and then we have a first speaker and a very enthusiastic, energetic scientist, Darshan Shah from Chemical Engineering, IIT Bombay, and he will be talking about the modeling mesenchymal stem cell behavior on viscoelastic substrate using principles of system biology. So he will be talking about this topic, and then this floor is open to Mr. Darshan Shah. Please take 15 minutes time is yours. Yeah. With that, I think we'll hand over to. Darshan.
Yeah, hi. Good afternoon, everyone. I hope you can see the screen.
Yeah.
So good afternoon, everyone. Welcome to my talk on modeling mesenchymal stem cell behavior on 2D viscoelastic substrate using principles of systems biology. Human mesenchymal stem cells, or HMSCs for short, are stem cells found in the blood and bone marrow, and they can differentiate to multiple connective tissue lineages like the bone, the muscle, and the fat. I have chosen to study HMSCs because of their widespread clinical importance in regenerative medicine and healthcare. Now, it has been known that this differentiation to multiple...
Sorry, I interrupt you, sir.
It has been known that this...
Yeah, tell me.
Sir, organizer asking for please not start presentation going on, sir.
Hello?
Please wait some time, sir.
Okay, who is telling this?
Sir, organizer side for my team, Mr. Nandan Bish, sir, asking for me.
Okay, okay, no
Please
problem.
wait some time, sir.
Sure, sure.
Okay,
I didn't know. Somebody told me to start. That's what I started. I have
Yes,
no
sir.
idea.
Yes, sir.
No
Sorry.
issue, no issue. No issue. No problem.
Dashaji, please wait some time.
Sure, sure.
Hello,
Hello.
sir. Plenary session is over, sir.
Over. Can we start?
Yes, sir.
Yeah. Okay. Thank you again. Once again, yeah. Welcome for the afternoon session. This symposium, PMRF Symposium 2022. I would like to thank the organizer for the opportunity to share the session. In this session, we have 5 speakers, and they're lined up, and they will be giving their talk 15 minutes each. The first speaker is Mr. Darshan Shah, right? Darshan Shah. He will be talking about the modeling mesenchymal stem cell behavior on viscoelastic substrate using principles of system biology. So he is from the Chemical Engineering Department of IIT Bombay. So Mr. Darshan will take another 15 minutes, and he will be able to speak for 12 minutes or 13 minutes. We can have more interaction on that question session, right? Darshan, take the stage. The stage is yours. Take care.
Yeah. Thank you, sir.
Yeah.
Just hi, I hope you all can see the screen. So good afternoon, everyone. Welcome to my talk on modeling mesenchymal stem cell behavior on 2D viscoelastic substrates using principles of systems biology. Human mesenchymal stem cells, or HMSCs for short, are stem cells found in the blood and bone marrow, and they can differentiate to multiple connective tissue lineages like the fat, the muscle, and the bone. I have chosen to study HMSCs because of their widespread clinical importance in regenerative medicine and healthcare. Now, it has been known that this differentiation to multiple lineages can be brought about by providing the cells with a suitable differentiating chemical induction media. But the importance of the mechanical properties of the substrate in driving this differentiation are just starting to be revealed in the last 2 decades. These HMSCs being adherent cells, they sense and respond to the mechanical properties of their microenvironment that they're plated on. If they sense a soft substrate, like one of a fat, they differentiate into fat cells and show a round unspread morphology. If instead they sense a stiff substrate, then they differentiate to bone cells and they show a spread morphology that is similar to these cells. Choosing which cell type to differentiate into for a given stiffness and many such cellular decision-making processes are regulated by something called as the mechanotransduction pathway. This complex series of biochemical and physical interactions begins at the focal adhesions, which are like the feet of the cells, and they are the cell's primary point of contact to the substrate. Hundreds of proteins, a select few of them listed here, are involved in transducing this extracellular mechanical information to the inside of the cell and mediating it into the nucleus, where finally changes in the gene expression generates appropriate cellular responses to these extracellular mechanical cues. Now, traditionally, cell biologists have studied a subset of this complex process via experimentation techniques to understand the mechano responses. But systems biology, on the other hand, provides a framework to study these complex systems from a holistic, big picture viewpoint and distill just emergent properties from the system which arise due to interactions in the network. So my goal is to utilize this novel approach to develop mathematical models, which would be useful tools to study this HMSC biology and understand the black box, which is the mechanotransduction pathway. Say, for example, the observation of cells plated on elastic substrates versus the cells plated on viscoelastic substrates. When you look at it on elastic substrate, the cell is nice and relaxed with no membrane fluctuations, whereas on viscoelastic substrates, there is an inherent uneasiness, a restlessness of the cell membrane. Another observation is that the focal adhesion or the cell's feet are very well developed in the first case, but not so in the second viscoelastic case. So we wanted to understand this underlying mechanism, as all biological materials are viscoelastic and they exhibit time-dependent dynamic mechanical properties. And cells which have undergone millions of years of evolution have tuned their cellular machineries to sense and respond to such mechanical properties. And there is very strong evidence that it drives cellular behavior in an altogether different way, as the table shows. For example, for spreading and proliferation, the behavior is kind of similar to what we see on a stiff substrate, but for other things, it is not so. So we wanted to discover a network that demonstrates such a unintuitive behavior. So to do that, we set out to develop an algorithm to quantify this wobbling observed. So the goal was to develop an algorithm that could automatically do this in an unbiased and reproducible fashion. So first, we developed an image processing algorithm in MATLAB that used Sobel edge detection algorithms along with pre and post processing of the images to finally extract the perimeter of the cell. From this perimeter, for different angular positions from the cell center, the radial distances were graphed with time. For example, here you can see that for 0 degrees and for 90 degrees. And the red line is that for the cell on the viscoelastic substrate, whereas the blue dotted line is the cell on the elastic substrate. So clearly the viscoelastic substrate leads to more deviation from this average radii. So this deviation was captured in a test statistic, which we termed as the root mean squared error, which gives an indication of the overall deviation of this radial distance from for all the different angles for one particular cell. And for multiple cells in set 1, which is the elastic substrate, and set 2, which is the viscoelastic substrate, we conducted a hypothesis test and we found that the difference was indeed significant. And this analysis has been published in 2021. And this work was done in collaboration with Pallavi, one of the colleagues from my lab. But still the question remains that what is the underlying mechanism? So currently our working hypothesis is that because of the loss of traction that happens because of the substrate creep in the system, the homeostasis is broken dynamically. There is a loss of isometric tension, which is the contractility forces, and which results in a simultaneous increase in the isotonic tension, which is the cell spreading forces. So the forces shift from the contractile to spreading forces. And the underlying theory which supports this hypothesis is the cellular tensegrity model, which says that the cell is made up of such tension-bearing and compression-bearing elements, which ultimately leads to a balance of forces, and hence the shape and the morphology is maintained. And when you can think of this situation as standing in quicksand, which is kind of difficult, and it requires the support of something of a large surface area to dissipate the unstable forces and maintain balance. This is what we are trying to do for a cell plated on viscoelastic substrate. And thus the aim was to develop a model that incorporates the force balance along with the biochemical signal transduction pathway. And so we took all the insights from the physics and using our heuristics from the systems biology, we developed a minimal mechanotransduction model that links the inputs of the substrate elastic and viscous moduli to the spreading area of the cell using a signaling pathway, a minimal signaling pathway, that is the logic, in combination with a forced transduction mechanism, which you can see in the red part of this figure. And it includes crosstalks, feed forward, and feedback loops, which are standard motifs in the systems biology framework. And we proposed that this is the minimal network that captures the wobbling behavior. Let us see how. First, let us consider the case of when the viscous modulus is 0 or near 0, which is very small. In this case, all the pathways associated with high viscous modulus are turned off, leading to a tensional balance achieved something like this. And we are interested in plotting the focal adhesions, which is the feet of the cells, and the spreading of the cells. So in this plot, as you can see, that if we simulate it with time, what happens is that when this viscous modulus component is not significant, with time these saturate and they attain a nice stable steady state. But now let us see what happens when the complete network is simulated and the viscous modulus is turned on. So because of the feedback loops and the crosstalks, there is a balance shift that happens in the system. And because of this shift in the balance, we see oscillations arise. So in the plot, you can see that even if the cell spreading is increasing, the focal adhesions are going down. And this is what we observe, that using our model, we can show that while the cell spreading increases, the focal adhesions decrease, and there is some oscillations that are reported. And lastly, this, of course, is a very simplified explanation of the whole mechanotransduction pathway. The real network is indeed very dynamic. But just to conclude, this minimal model helps us understand what is going on inside the mesenchymal cell decision-making process, and how the underlying network gives rise to such interesting phenomena. As you can see, running more simulations and playing with the model gives us many crucial insights, like spreading, oscillation observation, the focal adhesion dynamics, and many other features of the mechanotransduction network. I want to leave you all with my vision as a systems biologist working in this field. Apart from the substrate viscoelasticity, the cell integrates information from many other factors and then responds and takes appropriate decisions. So the vision is thus to combine state-of-the-art modeling and experimentation techniques to develop a multi-input, multi-output model that is good enough to replace the real system that works via chemical reactions with an in silico virtual platform that works using ones and zeros. This challenging and exciting field is at the intersection of systems engineering, mechanics, and cell biology. It is just waiting to be explored, and it has the ability to answer many open-ended questions in the field of mechanobiology and hopefully contribute towards our progress in healthcare and regenerative medicine. And with that, I would like to thank you for your time and attention. I would like to thank my guides, Professor K.V. Venkatesh and Professor Majumdar, for mentoring me through my journey. I would like to thank my lab members and the Department of Chemical Engineering at IIT Bombay and the PMRF Fellowship for giving me this wonderful research opportunity. Thank you, everyone.
Yeah, thank you, Darshan. One wonderful presentation. In fact, it is a very interesting presentation. And it is open for question, queries, comments from the audience. This presentation is now open. For that, I'll ask some questions. If anybody has any questions to be asked. Any comments from the audience? If not question? No. Okay. Darshan, well, good presentation, very good work. I have a few queries. From the real situation, real-time situations, how you will correlate with the cells? Have you taken up the consideration the real in vitro system or in vivo system? How you can extrapolate or something like that? Do you study?
Yeah. What we have proposed to do is that currently the model is at a stage where we are doing dimensionless equations and solving these equations using a dimensionless solver. The next step obviously is to correlate the data that we observe in the lab using the real data, and then use those correlations to fit the model to give an output for the real dimensional data. Like, for example, what happens for the spreading in terms of the micrometer square of the spreading area, or what happens to the focal adhesion concentration. So that is the next step that we are going to do. Currently, the model in its form is in a dimensionless state.
Okay, very good, very good. So when you do that thing, you take some cells. See, when you compare with the normal cells as well as the, your stem cells and normal cells, and then you take it for some cancerous cells, because they'll behave differently in different situations, right? So you take different steps, and then you consider for that also, cancer cells also, how they are behaving in this condition.
Yeah. So depending on the cell line that we are looking at, what we can do is we can change a few parameters in the model. And that would also give an insight as to what is different between the two cell types when you look at the whole mechanotransduction pathway. Let's say, for example, a cancerous cell behaves in a different way, but what is the underlying inherent difference in the whole transduction pathway, that we can find out using models like these.
Yeah, because see, the cancerous cells, they don't have the contact inhibition, right? They don't have, basically.
Correct. Yes,
so
yes.
that will maybe, I don't know, the parameter you have to accordingly, you have to modeling that way. That will help you, right? That will be good study, right? Very good. Yeah. Thank you. That's a great work.
Thank you, sir.
If anybody has any questions, and if no questions, then I thank the speaker, Darshan. All the best. Keep it, well done. Well done. Keep it up. Okay. Yeah. Thank you. And we move on to the next speaker. Are you ready? Next
Yes,
speaker
sir.
is.
Sir, I'm Himanshu Padwar from IIT Delhi.
Yeah, Himanshu. Welcome, Himanshu.
Yeah.
And now Himanshu, IIT Delhi, Electrical Engineering Department.
Yeah.
We'll be talking about some studies on graph signaling process with the application to Alzheimer's disease detection. A very good, interesting topic. Please, carry on.
Yeah.
Yeah. Interesting. Yeah. Yeah.
Sir, my screen is visible to you? Presentation?
Yes, yes, please go ahead.
Yeah.
Go ahead.
So good afternoon to all of you. I am Himanshu Padwar from IIT Delhi. First of all, I would like to thank the organizers for inviting me here to deliver a talk at this National PMRF convention. I completed my PhD in July 2021 from IIT Delhi in the Department of Electrical Engineering under the guidance of Professor S.D. Joshi and Professor Tapan Kumar Gandhi. And my research topic was some studies on graph signal processing with application to Alzheimer's disease detection. So now, as most of us know, Alzheimer's disease is a chronic neurological disorder affecting nearly 44 million people worldwide. It causes degeneration of brain cells, thereby resulting in loss of memory and cognitive decline, thus hindering a person's independent functionality. It is the most prevalent form of dementia. So, although serious efforts have been made to cure this Alzheimer's disease, the presently available medical treatments are unable to cure it reliably. However, detection of AD in its early stage, called mild cognitive impairment, is found to be useful in controlling or at least slowing down the progress of the disease. So many efforts have been made to solve this prediction problem using different techniques ranging from graph theory, network diffusion models to recently proposed machine learning and deep learning based models. However, the graph-based approaches in particular have gained significant attention in the recent years thanks to their ability to exploit the additional relational information between different brain parts. So in the present work, we first propose a graph signal processing based Alzheimer's disease detection model, which detects the Alzheimer's in its early stage, that is mild cognitive impairment. And then we propose a novel approach to coarsen a graph and also model its time evolving nature. And then both our proposed methods are then used to modify our earlier proposed Alzheimer's disease detection model to improve its performance further. Now the work carried here can be broadly categorized into three main parts. So the first is design of Alzheimer's disease detection model using graph frequency analysis of input brain fMRI data and a graph CNN classifier. The second is design of graph wavelet based multi-level graph coarsening algorithm with its application in AD detection. And the final part is characterization of time evolving graph connectivity dynamics using state space modeling with its application again in Alzheimer's disease detection. So now coming to the first part. In the first part, we design a graph signal processing based Alzheimer's disease detection model. And the work can be broadly categorized into two parts. So first is, in the first part, we propose a novel graph frequency based.
बीच में आपके म्यूट हो रहे हैं. Mike, not audible.
Sir, am I audible now?
Yes, sir, you're audible, sir.
Yeah.
बीच में बंद हो रहे हैं.
Okay, sir. Please stop your video, sir.
Okay. Just a minute.
Presentation, sir. Okay, sir, please. Slideshow mode, sir, please.
Yeah. Sir, is it fine now?
Yes, please go ahead.
Yeah. So in the first part, we proposed a graph frequency based feature extraction hypothesis, and it has been motivated by the results of two neurological experiments conducted separately, wherein by relating the findings of those two experiments, we proposed a hypothesis, a feature extraction hypothesis, that the high frequency content of a brain fMRI signal can act as a discriminating feature for early detection of Alzheimer's. Now, the features thus extracted are then provided to a graph CNN classifier, which acts as a graph, which is built using the graph convolutional neural network, which is extension of conventional CNN in the graphical context. So this is the block diagram of our proposed primary model. So here, first part is data pre-processing and graph construction. The second part is graph high-pass filtering, which basically extracts the discriminating features from this input fMRI data. So overall, these two represents the feature extraction part, and the features, graph signal-based features thus extracted, are then provided to a graph signal graph CNN classifier for the classification purpose. So this is the overall block diagram. Then coming to the results. So to compare the performance of our proposed model with the existing one, we compared the performance, classification performance, using three different measures of classification, which are accuracy, sensitivity, and specificity. So here are the results. So we can see this yellow one is our proposed model. So we can see in most of the cases, it outperforms all the existing methods. And this improvement in the performance can be attributed to two key factors. The first is obviously our novel feature extraction methods that extracts the highly discriminating feature from the input fMRI data using graph frequency analysis. And the second is in this model, instead of using the conventional machine learning tools like SVM, logistic regression, or CNN, we here designed our classifier using graph CNN, which is optimized for the irregularly structured data like the brain imaging data which is involved here. So Now coming to the second part. So in the previous model, we have used the ChebNet graph CNN architecture for the graph signal classification purpose. Although our previous model performed well and classified the normal control and the MCI patient successfully, in order to improve the performance further, we here modified the graph CNN classifier, which classifies the graph signal-based features. So similar to the conventional CNN, graph CNN also has two main operations. So first is graph convolution followed by the graph pooling. Although the substantial work has been done to improve the graph convolution part, the graph coarsening, which is equivalent to graph pooling, has received a relatively little attention and thus making a scope for improvement there. So in this work, we proposed a novel graph wavelet-based two-stage multi-level graph coarsening method, wherein in the first stage of coarsening, we propose we downsample a graph using a graph wavelet-based similarity measure, as the graph wavelets can exploit the additional local information and topological environment in the graph, in addition to extracting the direct edge strength, the information in the direct edge strength. And in the second stage, at each level of coarsening, we designed the reduced Laplacian operator as a restriction of original Laplacian operator to a dominant subspace, and which also simultaneously maximizes the topological similarity. And this formulation, this above problem was formulated as an optimization problem. So this is the flowchart of our proposed coarsening algorithm. So here, this L tilde is the result of first stage of coarsening, and LK is the final two-stage coarsened graph. And K indicates the stages of coarsening, means level of coarsening. And this is the pseudocode. So now coming to the results, we first compare our coarsening algorithm in the general coarsening context, and here are some representative results. So we compared our coarsening performance using different measures. One of it is shown here, that is operator dissimilarity index, and we can see that this bottom one is our proposed model. So it is performing best, because the lower the operator dissimilarity index, better is the coarsening method. So it is performing better than all the existing method. And then after verifying this, we applied it as a pooling operator in the graph CNN, and then used the modified graph CNN as a classifier in our Alzheimer's disease detection application. So this blue one shown here is the model with our modified graph CNN architecture. So here we can see that it is performing better than our previously proposed as well as all the other existing models. And this improvement in the performance can be attributed to our novel graph wavelet based method to downsample a graph, because the wavelets can rather than using only the direct extent, the wavelet can exploit the information in the local neighborhood and the topological environment also. And the second reason is that we at each level of coarsening, the coarsened Laplacian preserve the operator characteristics of the original graph Laplacian rather than preserving to the characteristics of the Laplacian at the previous level. So this may have reduced the overall information loss in the coarsening process. So in all the analysis, so now coming to our final part. So whatever we have discussed so far, we have assumed that the connectivity dynamics means the brain connectivity of the network remains static. That is, it does not vary with the time. However, the recent studies involving the dynamic brain connectivity has shown that there is altered brain connectivity as far as Alzheimer's disease detection is concerned. So it can act as a potential biomarker for Alzheimer's disease detection. So in order to exploit the information in the dynamic connectivity of a brain, we here propose a novel approach to characterize the connectivity dynamics of a time-varying graph using state space model. So here is our proposed model. So we model the instantaneous graph connectivity as the state of the model, this AK, and then the input graph signal at time k is modeled as an observation or measurement vector. So this SK is the input graph signal. And now this state transition matrix F essentially captures the connectivity dynamics of a time varying graph. State transition matrix. So it acts, this matrix F acts as a feature for Alzheimer's disease detection. So this is the block diagram. So first is brain parcellation and graph construction. So this is pre-processing. Then state space representation. So it gives us the matrix F, which is the feature for AD detection. And then this matrix F is given as an input to CNN classifier for the final classification purpose. So we once again verify the efficacy of this model, dynamic connectivity based model, in our present Alzheimer's disease detection application. And after verifying that, in order to improve the performance further, we combine both this dynamic connectivity based model with our earlier proposed static connectivity based model using multimodal deep learning. So here we use the intermediate level multimodal deep learning approach. So we can see the first one using graph CNN is the static connectivity based model, and the bottom one is showing the dynamic connectivity based model. It is using conventional CNN, and the features extracted here, FC static, that is fully connected layer of this model, static based model, is concatenated with the fully connected layer of dynamic connectivity based model. Okay, and then this joint feature representation is given for the final classification purpose. So now here are the results. This is for the dynamic connectivity based model, and this right one is for the integrated model using both the dynamic and the static model. So this red one is the obtained results by our model, and here this yellow one is showing the results of our proposed integrated model. So here we can see the improvement, and this improvement can be attributed to our novel method to characterize the connectivity dynamics of a time varying graph using state transition matrix of our SSM based model, as it encodes intrinsically the connectivity dynamics of a time varying graph, in contrast to the existing method where they use the instantaneous graph parameter values to characterize the connectivity dynamics, and the parameters are often chosen empirically. And the second reason is our integration of static and the dynamic connectivity based model, as they can as it can exploit both of the features simultaneously. So I think the slide has stopped working.
Please click on center, sir.
Yeah. Okay. So that was all. Okay. These are some of the publications based on this work, and these are the references. Thank you all. Thank you to all of you.
Okay, Himanshu. Well presented. Very good. Good work, Himanshu.
Thank you, sir. Thank you.
And now this presentation is open for discussion. Any discussion from the comments from the audience? Welcome. So, hello.
Yeah.
Nobody is there. Any questions from the audience side? Okay, Himanshu, it's a wonderful work. I have a question. So this Alzheimer's disease, how early it can be detected with your model? How early?
How early is in the MCI stage, sir? So MCI stage typically comes around 5 to 6 years, means earlier than the Alzheimer's stage.
Okay. So,
if you are means getting Alzheimer's after 5 to 6 years, then the MCI stage comes earlier to around 6 years. So we can have that much of means.
so 5 to 6 years earlier you can detect with the
Yeah,
help of this,
yeah,
your model.
Yeah.
Okay. So have you ever conducted with the real model?
Sir, I have worked with the ADNI dataset. So ADNI is the standard dataset as far as Alzheimer's is concerned. So they have labeled both type of subject, that is normal, MCI and Alzheimer's. So MCI specifically are the subjects which are having the initial stages of Alzheimer. So we have worked on that data set.
I see. Okay. Very good. Good. So this is always you take the data from the MRI and fMRI and right?
Yeah.
Any other method to you can adopt from this other than MRI?
Other than MRI,
CT you can use CT and all other. No.
Sir, but data set means standard data set is in terms of fMRI. So we worked with fMRI. Means, no data set was available in other modalities.
Okay.
So we have not tried on the means on other modalities.
So this is available data now.
Yeah.
Good work, Himanshu. Keep it up.
Yeah.
So you have completed, right? This PhD work you
I
completed.
completed in July 2021.
So good. All the best for your future.
Yeah. Thank you.
Best of luck. Yeah. So that end of the second speaker, and we are moving to move on to the third speaker. I hope third speaker is Paulomi Banerjee. Are you there,
Yes.
Paulomi?
Yes, sir.
Yeah.
Good afternoon, sir.
Good afternoon. Paulomi is from Materials Engineering, IISc Bangalore. She will be talking about tailoring the interface between epoxy and carbon fiber laminated system through self-healing routes. So, follow me. You take the stage is yours for 15 minutes. You take. Yeah.
Thank you, sir. So good afternoon to respective dignitaries. My work is primarily on
Please share
the...
your screen.
Yes, is my screen visible?
Not yet, ma'am. Please share your screen.
Not it.
Just a second. Yes, is it visible now?
Yeah, that's fine.
Yes, please
put it the presentation mode.
Yes. Yes, is it visible? Is it fine?
Yeah. Fine.
Okay. Okay, so my work is primarily on the interfacial engineering of carbon fiber reinforced epoxy laminates, or widely known as CFRP laminates, for their mechanical property enhancement and self-healing ability. So the system that I work with is laminated system, which has two components: epoxy as the matrix and carbon fiber mats as the reinforcement. And such CFRP laminates are widely used in aircraft industries. So the origin of use of CFRPs in aircraft started from around 1970s, and during that time, only 10% of aircraft components were fabricated using CFRPs. But now, aircraft such as Airbus A350 have around 52 to 60% of its structural components fabricated with CFRPs, mainly, primarily the wings and fuselage of such aircrafts. This is mainly because CFRPs have an extremely high specific strength and extremely high specific modulus, and their properties can be very easily tuned. The major problem with such laminated systems is that since these structures are widely anisotropic in nature, because of the difference in thermal expansion coefficient between the carbon fibers and the epoxy, there exists a weakened interface, which subsequently leads to interfacial delamination and is a major cause of failure in aircrafts. Furthermore, there is always a trade-off between enhancement of in-plane and out-of-plane properties, because any modification at the interface always leads to a deterioration of the fiber-based properties. Next, very little to no research work has been conducted on the self-healing, interfacial self-healing ability of such aircraft structures on such composites, because whatever research has been conducted on these composites has been through microcapsular or vascular mode of self-healing technology, and this always leads to a degradation of the original mechanical properties. So my objectives are primarily two. Firstly, I have planned on enhancing the interface of CFRPs to substantially improve the matrix adhesion over fibers, and thereby improving both the in-plane and the out-of-plane properties, which is through improvement in flexural strength and interlaminar shear strength or ILSS properties. Secondly, I have also devised a self-healing technology at the interface in an intrinsic method, which would maintain and even improve the original mechanical properties and lead to considerable strength recovery post-failure. So the fabrication protocol for my laminates is through VARTM, or vacuum assisted resin transfer molding. Now, this is a widely commercialized process for fabricating CFRPs, and it's a fairly elaboration fabrication technique. So it has taken me firstly several months to master the art of VARTM fabrication. So this is mainly how the fabrication goes. We have 10 layers of carbon fibers. Yes, so for my system, I have fabricated laminates with 10 layers of carbon fibers, inside which epoxy is infused in each subsequent layer, and a vacuum bag is placed over this over my laminate with the resin infusion tube at one end and the vacuum pump at the other end. Now, when the vacuum pump is started, it is through the capillary action that epoxy is infused and distributed properly over the fibers. Following the laminate fabrication, curing and post-curing cycle is conducted to which leads to cross-linking of my epoxy, which is a thermosetting polymer. And after that, to conduct the mechanical test, my 15 cross 12 centimeter square laminates are cut according to the respected ASTM standards. So the first method in which I have devised an intrinsic interfacial self-healing technology in my laminates is through a thermoreversible covalent bond formation, or a Diels-Alder bond formation at the interface. Now for this, I have modified my epoxy matrix with graphene oxide. Now graphene oxide has conjugated diene bonds at its edge. And I have covalently functionalized my carbon fibers with bismaleimide. So bismaleimide has dienophilic components. So the diene and dienophilic components can together create a Diels-Alder adder. The covalent functionalization has been done so that during the infusion of epoxy into my laminates, the organic moiety is not washed away. We could see after subsequent mechanical testing that both the flexural strength and ILSS values considerably improved as compared to the unmodified laminated structures, which showed that a much more strengthened interface was created. Following the self-healing cycle, when further ILSS test was conducted, we could see a considerable recovery of the interfacial strength, which was around 65 to 70% of strength recovery, implying that intrinsic self-healing had worked. So here in the SCM micrograph of our fractured samples, we can see that while the neat carbon fiber epoxy laminates show barren fibers, just plain matrix modified epoxy carbon fiber laminates show a higher adhesion of epoxy matrix over the fibers, implying that presence of graphene oxide led to a stronger interface. Finally, For the laminates in which both the matrix and the fibers were modified, we could see that the epoxy resin was very nicely adhered over the carbon fibers, which can clearly confirm the fact that the interface was properly strengthened and the interfacial properties were considerably improved on the subsequent modifications. Now, however, it must be told that carbon fibers suffer from structural disintegrity on chemical treatments, and thereby for my next work, I tried a non-chemical or ionic form of fiber treatment routes on the fibers to lead to improvement in interfacial properties, so without degrading the original fiber properties. So for this, in my next set of work, I designed two forms of architecture in which I in situ grew zinc oxide nanorods on the carbon fibers. Now, zinc oxide nanorods provide a through thickness matrix anchoring form of rigid mechanical interlocks through a zipper-like technique, which can form interlocks over large areas of the matrix and thereby improve the interfacial properties. Also, since zinc oxide nanorods are rigid in structure, there is even less propensity for this to be washed away on the resin infusion during the VRTM process. So for my next set of work, the two forms of architecture that I developed, one of which contained 10 layers of carbon fibers, each of which had zinc oxide nanorods in situ grown with my epoxy matrix modified by graphene oxide. In the second form of architecture, I had alternate layers of zinc oxide nanorods grown carbon fiber alternated with bismaleimide covalently deposited carbon fibers. So my second structure was devised in such a manner which would subsequently considerably improve the mechanical interlocking of the laminate system and also provide an intrinsic self-healing technology. So to in situ grow ZNO nanorods on the fibers, I followed a CLAR form of technique, which is successive layer adsorption and reaction process. In here, there is a seed coating phase in which zinc acetate seed is deposited on the fibers, followed by a growth phase in which zinc nitrate salt, which with HMTA is dipped into the fibers and taken and kept at a temperature of 90 degrees centigrade for uniform growth of ZNO nanorods on the fibers. So SCM micrographs can show that after optimization of the time, temperature, and concentration of my moieties, zinc oxide nanorods were uniformly grown on the carbon fibers. Now, both EDX and XRD showed that the zinc oxide nanorods that were grown were of woodside structure, which means that they were hexagonal crystalline ZNO. Now, I conducted both flexural and ILSS analysis in similar way as I previously mentioned. And in here, we could see that again, both the flexural and ILSS properties of my laminates were considerably improved in comparison to that of the or the unmodified CFRP laminates. Following the self-healing cycle, it could see that there was an even better strength recovery of this laminated systems in the laminates, which had alternate layers of bismaleimide deposited carbon fibers. So this could be attributed to several things. Firstly, when we conducted an epoxy contact angle analysis on our bare carbon fibers, we could very easily see that as compared to the epoxy droplet forming on the neat carbon fibers, modified carbon fibers showed a decrease in contact angle, which meant that there was higher surface adhesion. Furthermore, the fractured samples showed a much better adhesion of epoxy over the carbon fibers. As you can see that carbon fibers in this fractured samples are not quite visible at all, which means that epoxy has very nicely wrapped around the carbon fibers. And on closer inspection of through SCM micrographs, we could see that the ZNO nanorods were intact over the fibers. And not only that, we could also see that epoxy had adhered over ZNO as well, which meant that the ZNO was providing an even higher surface area and higher surface energy for epoxy to be wrapped around, subsequently improving the interfacial strength. So from here in terms of values, we could see that as compared to the neat CFRPs, the bismaleimide CFRPs, where graphene oxide of 0.28% was added in the matrix, there was a 30% improvement in the flexural strength and around 43% improvement in ILSS values. Also, in samples with 0.58% in ZNO BMI CFRPs, which had alternate layers of ZNO carbon fibers and bismaleimide carbon fibers, with matrix modified with 0.58% of GO, there was a 46% improvement in the flexural strength and 33% improvement in ILSS properties. And this considerable level of improvement has been has not been reported in current literature to the best of my knowledge. Also, this intrinsic form of self-healing technology, which is very novel in my knowledge, has led to an improvement of, has led to a self-healing efficiency of around 65 to 70%, which is a considerable improvement in strength recovery, which would subsequently lead to an increase in the durability of such structures. So up until now, I have fabricated several hybrid CFRP structures, which I mentioned. The first one was that of neat CFRP, in which the interface is unmodified. In the second case, I have fabricated CFRPs with geo modified in the geo dispersed in the matrix, and some of these geos would sit at the interface because of interaction with the carbon fibers through pi-pi interaction. In the third scenario, a Diels-Alder bond has been generated in the interface using a thermoreversible reaction between the fiber and the epoxy. In the second approach, we have went into a more of a mechanical interlocking sort of technique, in which carbon fiber has been modified with ZNO nanorods grown in situ over the fibers, and a mechanical impingement or through thickness technique is how the interface has been strengthened. In the fifth form of fifth architecture that I have devised, I have created a synergism between mechanical interlocking and covalent or Diels-Alder form of interfacial strengthening, which would not only improve the properties of the interfacial properties of the laminates, but would also lead to subsequent strength recovery via an intrinsic form of self-healing technique. So further, for the next rest of my PhD, I plan to devise another modified CFRP laminates in which there is a site selectivity of deposition of bismaleimide over ZNO, so that bismaleimide can interact not only with the interfacial graphene oxide, but with the graphene oxide present in the bulk of epoxy as well, which I believe would consequently improve the self-healing ability of such structures to around 90 to 95%. So as I mentioned, the next in line work in my research involves fabrication of novel modified CFRP laminates with site selective bismaleimide anchored over GO over ZNO, and assessment of both the flexural and ILSS properties of such structures. Furthermore, I also plan to work on an in-service mechanical behavior such as fatigue analysis, because fatigue analysis gives us a more predominant idea on how the self-healing technology would work in real-time applications. So these are the publications of mine up until now. And finally, I would like to thank my supervisors and my laboratory colleagues for their help and assistance. The Department of Materials Engineering in Indian Institute of Science, Bangalore, for providing me with the necessary resources, PMRF Fellowship, and the Ministry of Education for organizing this esteemed symposium today. Thank you.
Well presented, Olomi. Good. You keep it in
Thank
time.
you, sir.
Good presentation. Good work. So now this is open for question answer. Anybody else from the audience side? Any questions? If there is no questions, I have a few queries. Follow me. What is the size of the particles you used? Did you check the size of the nanoparticle? This carbon?
Yes, sir. Graphene oxide particles are around 30 nanometers in dimension, and I disperse them well enough so that any agglomeration or accumulation is taken off. So after that, when I have done the SEM, I've seen that the size of graphene oxide is around 30 to 32 nanometers.
But the shape, the rod, or what is the shape?
Okay, so zinc oxide nanorods have a dimension of like, we analyzed the ZNO using Debye-Scherrer method, and we saw that they have a size of around 26 to 28 nanometers in cross-sectional area, while the length is around 1 to 2 micron.
The healing is depending on the size or the strength of the layer. Which
So
is
the
better?
healing, we believe that the self-healing is dependent on the amount of bismaleimide on the interface and the amount of graphene oxide which can migrate to the interface. So we do hit a saturated layer, after which the interface becomes little more viscous with the migration of GO, so that again imparts the adhesion of epoxy. That would again lead to decrease in mechanical strength. So we have to basically hit a sweet spot in terms of concentration.
Okay. Did you compare with the normal without the epoxy and without your particle treated material and this one? What did you check with that, the control one?
Yes, so the controlled samples need CFRPs when we subject them to self-healing without any modification. The samples fail very early. Like when the stress strain graph is plotted, the test is conducted, the samples fail when at around just 100 to 150 Newton. It does not reach higher. But when modifications are done using self-healing, we can see that the slope, the plot, the graph is keeps on going up to a higher range of force or higher range of load.
Okay. It's a resistance power is more, right? This is healing power.
Yes, sir.
Is material used in the aeroplane or any other place it is used?
CFRPs are widely used in aircrafts these days. We have Boeing 727, Airbus A350. This aircraft use CFRPs in their wings and fuselage. But the research on self-healing of laminates has not been done up until the last 5 years or so. So it's a very novel field of work.
Where it can be proper scope of application, this material, other than aircraft?
Since I'm using VRTM form of fabrication technique, VRTM is widely used for aircraft or spacecraft technologies, or rockets and space shuttles as well. So in any sort of such structural component application, this laminates can be widely used.
Can be used. Okay, very good. Good work. Keep it up.
Sir,
all the best.
1 2 question on Q&A box, please check. Q&A box, please check.
Hello?
Hello?
Yeah.
Sir, 1 2 question on Q&A box, please check.
By chart path. Okay.
Yeah, question and answer, sir.
Where you have to check it? I don't find it in the
Sir,
chart.
question and answer, sir.
Okay. This is from Professor Vaidali V. Srikanth. What is the motivation behind choosing ZNO? Number one. Second one is the enhancement, enchantment is mechanical properties correlated with the spectroscopic analysis, Raman and SPS.
So this question is in 2 parts. So to answer the first part, we have seen several cases in which nanostructures or nanowires such as that of CNTs are grown on carbon fibers using CVD processes, CVD or PVD processes. However, such nanorods can be easily washed off during the VARTM process when epoxy is infused. That's why I wanted to develop a much more rigid nanostructure. Furthermore, ZNO, because of its stiff nature, provides a better mechanical interlocking than the consequent CNTs or graphene oxides. So this was the major reason why I chose ZNO, and also the the synthesis technique of ZNO on carbon fibers does not lead to any subsequent degradation of fibers, because carbon fiber degradation takes place at a very ambient temperature. If you heat the fibers to around, say, 150 or 200 degrees centigrade, the fibers will start disintegrating. So I wanted to incorporate nanostructures in such a way where the fibers' original properties were retained. That is why I chose ZNO. Now, the second point is whether the enhancement in mechanical properties can be correlated on spectroscopic analysis, Raman and XPS. Through XPS, so this work is, manuscript is currently under preparation. I have also done the XPS analysis of this. I could see that through XPS analysis that there is an ionic bond formation between ZNO and the COOH present on the surface of carbon fibers. And through Raman analysis, I could see that the ID by IG ratio of the ZNO modified carbon fibers increased. So this increase in disordered sites on the surface of carbon fiber led to more activity on the surface of carbon fibers, which subsequently led to increase in surface area and surface energy. And this higher surface energy is what leads to higher affinity for epoxy to wet the fibers.
Okay. I hope it is answer to that question. And she has same thing in other words, can we estimate mechanical properties through the spectroscopic analysis? This is what she's asking.
Yes, so as I already mentioned that we can conduct several spectroscopic analysis which can give us a rough idea on the wettability of the matrix over the fibers.
Okay. The other question is, Ms. Sumanna Gupta, can you throw some light on the cost difference?
Yes, so we have not performed any cost analysis on our work because we are at a very preliminary stage of our work where we can see whether interfacial engineering can lead to self-healing properties. But in subsequent years of our work, when translational research does take place, we definitely plan on seeing the impact of this on the cost difference.
Okay. I think that's all. Any more questions? Then there's questions only. I could see. Wait a minute. One more question. Yeah, that's all. Thank you all.
Thank you, sir.
Thank you, Manami. Well, good work. Good defining the answers, question answers. Good. All the best. I think we can move on to the next speaker. Probably the next speaker is ready.
Yeah.
Next speaker is Kandesh Agarwal. Are you there?
Yes,
Kandesh Agarwal ji? From chemical engineering, IIC Bangalore again. So he will be talking about inkjet printed nanostructured electrodes for PEM fuel cell. So Kandesh Agarwal ji, he will be giving the 15 minutes presentation now. Yeah, please.
Are you able to hear me properly? And I'm
Yes, yes. Yeah.
My presentation is in the full screen mode.
Fine,
Are you able
fine.
to see that?
Yeah, we can.
Okay. So thank you very much, Professor Rajamani, for the introduction.
Yeah.
Hello and welcome everyone to my talk entitled Inkjet Printed Nanostructured Electrodes for PEM Fuel Cell. Hydrogen is considered as fuel for future because it has got the highest calorific value among all the fuels known so far, and it can be produced from varieties of sources ranging from reforming of the natural gas to the electrolysis of water. There are two ways to extract energy out of hydrogen. The first one is the conventional combustion route, where hydrogen reacts with the oxygen in the gaseous phase to form water. In this case, molecular level electron transfer takes place, and because this process is very fast, the released energy is in the form of heat, and that is why it is very low efficient. Contrary to that, what is cleverly done in the fuel cell is that the hydrogen gives off its electron on an anode catalyst, which is generally a platinum-based catalyst, and the released proton is made to traverse through a proton exchange membrane. This is a selective membrane which allows only the passage of protons and not the electrons. So the electrons have to traverse to an external circuit, generating electricity. In this case, because the potential energy of the electrons is extracted as an energy, this is much more efficient than the conventional route. To incentivize the hydrogen economy, Honorable Finance Minister has recently announced 800 crores budget for National Hydrogen Energy Mission. And in that context, several organizations have come forward to develop fuel cell-based vehicles. One of the recently developed, indigenously developed fuel cell bus is by Sentient Labs in collaboration with NCL Pune and Sikri Karai Puri. And this is an important step towards Atmanirbhar Bharat. IOCL and Tata Motors are also trying to test the operability of the fuel cell bus on the streets of Delhi with the hydrogen network that they have in the IOCL R&D center at Faridabad. However, one of the major bottlenecks of mass adaptation in the fuel cell vehicles is the high cost associated with them. A cost comparison of a representative gasoline vehicle and a fuel cell vehicle indicates that the fuel cell vehicles are around 2.5 times more costlier than the gasoline vehicle. And in part, this high cost is associated with the high amount of platinum loading that goes inside a fuel cell catalyst compared to the gasoline vehicle catalytic converter. More so, the platinum cost becomes even more challenging when the number of fuel cell systems are increased per year. And this is very important if we have to go towards hydrogen economy by scaling up the number of fuel cell vehicles. The cathode side structure of a PEM fuel cell is quite complicated because proton from the proton exchange membrane, electron from the external circuit, and oxygen from the cathode side have to meet on the three-phase boundary. And therefore, the cathode side catalyst has to be conducting enough for the electron to be able to reach to the catalytic side. It has to be porous enough for the oxygen to diffuse, and it has to be hydrophilic enough for the water to leak away. And all these characteristics should simultaneously exist in the catalyst. And the current best possible compromise available is in the form of carbon cloth. And because it has a low conductivity, it has to be sufficiently thick, around 250 microns, which lead to mass transport loss, especially at the higher current densities, where a lot of water is generated, and that water impedes the flow of reactant and product transfer. So that is why a thin film along with the ultra low platinum loaded catalyst is desirable. Looking back at the evolution of platinum electrocatalyst for PEM fuel cell, it has evolved from using around 10 grams of platinum in the form of platinized platinum or platinum black in 1970s, where it was restricted to majorly space application, to around 2000s, where platinum nanoparticles were coated on carbon microsphere. This approach could reduce the platinum loading to 300 microgram per centimeter square. And recently, researchers have tried to alloy the platinum nanoparticles with the transition metals such as cobalt, nickel. This increases its activity. But the problem with the carbon-supported catalyst is the corrosion of the carbon base and inaccessibility of some of the platinum clusters, as indicated in this transmission electron microscopy image. The degradation of MEA from Toyota Mirai also indicate that the catalyst layer reduces its thickness from 9 microns to around 3 microns over the course of its life. So clearly, durability is one of the key issues in the fuel cell vehicles. One of the ways to avoid the carbon corrosion is developed by company 3M, and it's called as the nanostructure thin film electrode, or NSTF. This can reduce the platinum loading to 50 microgram per centimeter square. In this approach, they are able to get the complete coverage of platinum by thermal evaporation on the organic crystals. However, this approach also uses the vacuum deposition, which is not additive in nature, and it's a batch process, which is not very conducive for scaling up. One of the scalable approach is reported by Professor Moffat's group and called as the self-terminating electrodeposition, or STED from now on. This can reduce the platinum loading even less than 50 microgram per centimeter square, and can give platinum in the thicknesses of less than 10 nanometer. This approach is being pursued by Toyota R&D, and this has been shown to get translated to different substrates. So clearly, non-corrodable catalyst synthesis using scalable approach is the need for hour. In this context, we pitch in the idea of forming additive, roll-to-roll, and low-cost process for synthesizing carbonless PEMFC electrocatalyst. We are using an innovative print expose developed process to get the conducting as well as porous nanostructures to replace the carbon backbone in the catalyst. We are developing these structures directly onto the proton exchange membrane or Nafion membrane to avoid the complex ink preparation method or the decal transfer method. Further, on account of their being electrically conductive, we are using them as a template to deposit platinum as thin atomic layers with the help of STED method. This is an innovative process by which you can deposit platinum in an atomically layer by layer fashion just by controlling the potential between two values, which is -0.8 and +0.4 volt versus saturated calomel electrode. And depending on the number of potential cycles that you give in, it will have the number of platinum layers. So essentially, we aim at developing platinum atomic layers coated silver nanostructures, which look something like this. So let me give you a brief about what is the figure of merit in a PEMFC electrocatalyst. So if my electrocatalyst is electrochemically active, then I should see its signature corresponding to the proton reduction and oxidation in the acidic solution. So this is done in a simple 3-electrode chemistry where using the working reference and counter electrode in the acidic solution. The figure of merit here is shown with the parameter called as the ECSA, and the hashed area represents that. The US Department of Energy has a durability target that the electrocatalyst should not lose more than 40% in the 30,000 cycles, which is a typical lifetime of a fuel cell. So in initial phase, we found that there are several important parameters that one should take into account while measuring the ECSA or conducting the durability analysis, namely what electrolyte you should be choosing, how you should be choosing the baseline, what is the implications of potential scan window, what are the effects of dissolved oxygen, as well as organ impurities. So we have done a thorough literature analysis on the effect of ECSA measurements by these parameters and recently published a review article. So the question that we wanted to answer was how many platinum cycles of steps we require to form a durable electrocatalyst for a fuel cell vehicle. And to answer that, we have done a controlled study where we have deposited different number of cycles ranging from 1 to 20 on gold thin films which were e-beam evaporated by switching the potential between these two values. We estimated the platinum thickness with the help of an approximated model from XPS as well as step height measurement from the AFM, and both of them confirm the thickness of less than 5 nanometers. We measured the platinum loading with the help of ICP OES, which confirmed the less than 20 microgram per centimeter square platinum loading. We further subjected our samples to the XC2 durability analysis between 0.65 and 1 volt. These are the typical potentials that a fuel cell vehicle will experience. And the idea is that we are going to simulate similar kind of situation in a simple 3-electrode setup. And we will measure the activity at an intermittent level of these durability analysis. What we found was that for 8 or more cycles of platinum, we could achieve less than 40% ECSA loss. And the ECSA values for these durable samples was around 11 meters square per gram, which was close to what is given by the commercially available one, which is 14 meters square per gram. However, what we could achieve was our platinum loading in the case of durable samples was less than 10 microgram per centimeter square, which was 30 times lower than that of the commercial one. And the thickness was less than 5 nanometers, which was roughly 1,000 times less than that of the commercial one. We are well below the DOE limit of platinum loading. Further, we wanted to translate the capability of synthesizing these platinum structures onto the silver nanostructures, but instead of directly jumping onto the nanostructures and to avoid the complications associated with them, we have taken the commercially available silver thin films and started doing the platinum deposition and observing its electrochemical activity. We anticipated the challenge of silver plus formation because unlike controlled study where we have used gold, silver is not an inert substrate. So we modified our deposition protocol accordingly to prevent the galvanic displacement and localized fitting corrosion. As you can see that this is no more a two-way cycling, it's just one-side potential. Further, we also activated the samples to leach out the extra silver by sequentially increasing the upper side potential limit in the cyclic voltammetry till the time our CV starts to get the platinum looking like structure. And these activated samples were durable enough to retain 85% of the ECSA. Here too, we could get the durability, required durability with just 70 micrograms per centimeter square loading, which is almost like half than that of the US DOE target. We characterize these samples with the EDS and XPS to understand the surface composition of the thin film, and both of them gave the platinum to silver ratio to be around 2, which indicates that there might have been a stable and thin film formation of platinum and silver, which is able to give this exceptional durability. Further, we have taken the inkjet printing technique to develop the conducting and porous nanostructures on Nafion membrane. This technique was developed by our group, where we have combined the black and white inkjet printing with the silver halide-based photochemistry, and we are able to generate these nanostructures on different substrates like papers or membranes, etc. And this uses a very simple inkjet printer, which is just 2,000 rupees. The FESN image of silver nanostructures showed the porous nature and the thickness of such films can be less than 1 micron. This technique is an extension of the calotype process reported by Henry Talbot in 1841, and it was a precursor to the digital photography technique developed by Kodak and Canon. We have started doing the performance analysis of platinum deposited on these nanowires, and we were able to get the durability only till 1000 cycles so far. The reason of such low durability so far can be that they're not able to retain the morphology of these nanostructures. As you can see that after durability analysis, the nanowire, the underlying nanowire is breaking into the bulges or the nanoparticle kind of morphology. So we have modified the underlying nanostructures and we have started doing the deposition on the silver nanoparticles. The beauty of our printing process was that just by wearing the halide, we could synthesize, instead of wires, we could synthesize particles. And even with the nanoparticles as base, we are able to get the electrochemically active platinum. What now we have realized is that the loading of the silver also will matter a lot, because that is the conducting backbone, and depending on the loading, the characteristics of the platinum will be visible. So in future, we will be doing the activity and durability optimization for platinum deposition on silver nanostructures by optimizing the amount of silver as well as amount of platinum along with the electrochemical activity and electrical conductivity. And further, we will be doing the in situ performance testing, where we'll be using actual pressurized hydrogen as well as oxygen conditions. So in conclusion, I demonstrated the proof of concept for synthesizing the durable platinum overlayers using the STED approach. I explained to you how we could synthesize durable platinum atomic overlayers, which were 30 times lower platinum loaded with less than 10 microgram per centimeter square and less than 5 nanometer thickness, which is 1000 times lower than the commercial ones, and yet they were durable as per the fuel cell vehicle requirement. Our results with the thin and silver thin film showed the exceptionally durable platinum film, because we have done the appropriate modification in the protocols for deposition as well as activation. And our initial results with the silver nanostructures hold promise for synthesizing ultra-low platinum loaded catalysts for high surface area nanostructure substrates in the case of for the fuel cell vehicle application. I would like to thank our funding agency SCRB and our collaborators ONGC Energy Center Trust. I'm also grateful to having received the Prime Minister's Research Fellowship. With this, I conclude my presentation and I'll be happy to take questions. I can further be contacted on the email address given here. Thank you.
Well presented, Kandesh. Very good presentation. Good. Can you hear me, Kandesh?
Yeah, I can.
Good presentation. Now this presentation is open for questions. Anybody have questions? Either you ask, unmute yourself and ask, or you can type. Kandesh, good presentation, and I wanted to ask you, what is the, you are talking about the durability and the longevity. You are comparing with the platinum and the other one. No, what is the durability you are expecting from this maximum? Compared to others?
Correct. So the commercial ones are durable till 5 to 10,000 cycles. But the guideline says that it should be durable till 30,000 cycles, which is a typical lifetime of a fuel cell vehicle. So what we have shown is that our samples were durable till 30,000 cycles, meeting the US DOE targets.
30,000.
Yeah. So the 30,000 is decided based on how much, how much an average fuel cell vehicle can last.
Okay. If you change the other particles, like there are so many other particles are available. Did you try with other than silver or any other particles?
No, the idea is that we wanted to use the conductive and porous network of the silver. We haven't tried with the other ones. So the idea is that because they are in the nano regime, they have sufficiently high surface area as well as they are porous enough. So we wanted to try with that as a substrate. And silver is known to have the highest conductivity among all the metals.
Okay. Now we are talking about the in situ studies. How you do that in situ? What do you mean by
No, that in situ testing is in the actual fuel cell conditions where you will have the pressurized hydrogen and pressurized oxygen. So the studies that I have shown are done ex situ. That means they are in the three-electrode setup where you have acidic solution. It's a standard three-electrode chemistry. So these studies are the initial screenings, and after the satisfactory performance in the ex situ, you go for the actual performance, which is in situ. Because in situ requires quite a lot of involved preparation of membrane electrode assembly and stuff like that. So these are kind of initial screening tests for any electrocatalyst.
Okay, very good. So there is a question from Professor Vaidali Srikanth.
Yes.
Is there any possibility of replacing platinum with any other material such as metal oxide, which I asked? Yeah.
Correct. So yeah, people are also interested in synthesizing what is called as a non-PGM, non-platinum group metal catalyst. But platinum is by far known the best catalyst which can withstand the acidic conditions that are there inside the fuel cell environment. Of course, it has the highest activity for the oxygen reduction reaction as well as the hydrogen oxidation reaction.
Okay. I hope your answer to that question, Dr. Professor Vaidali. Okay, if there is no question, then we'll thank the speaker and we move on to the next speaker. Our next speaker is what is his name? Madhur Prashar, right? Medical Science and Technology, IIT Kanpur, Kharagpur, I'm sorry.
Yes, sir.
Development of magnetometry based single neuron resolution functional imaging, right? So, Madhur Prashar, sir. Yeah, please take whether 15 minutes is yours.
Hello everyone, I'm Madhur Parashar, PhD student at School of Medical Science and Technology, IIT Kharagpur. The title of my talk is Development of Magnetometry-Based Single Neuron Resolution Functional Imaging. This work is supervised by Dr. Shobha Bandyopadhyay, Dr. Kasturi Saha, and Dr. Sanjay Bandyopadhyay. So a brief. So initially, in principle, I would like to explain the core motivation for the project. We are trying to develop a novel technique for imaging cortical spiking activity in mammalian neuronal circuits. So in principle, this technique should look like we access the 3D neuronal cortical tissue through a cranial window. In this window, there are 3D neuronal, three-dimensional volume of neurons which have these action potentials. These will produce ultra small magnetic fields of the scale of picotesla. These ultra small magnetic fields are going to be detected by a bunch of electron spins hosted in a diamond crystal. The state of these spins are detected by an optical microscope, and the states are perturbed by applying a microwave perturbation. There are two main parts of the project. The first is which addresses how do we solve the inverse problem of reconstructing the three-dimensional neuronal activity from two-dimensional measurements. And the second is to actually develop this quantum diamond microscope, which is able to probe these ultra-low magnetic fields at microscale resolution and at room temperature. So let us consider what is the current state of art in neuronal imaging. The most important technique is functional two-photon calcium imaging, with the specifications that it can resolve single neurons. It is diffraction limited resolution. The temporal resolution is of the order of millisecond, but the key limitation here is that it can only access areas of the brain that are 1 mm from the brain surface. So why does the diamond nitrogen vacancy defect-based centers come into picture? First, they are room temperature ultra-sensitive magnetometers. They can come into close contact with brain tissue as they function at ambient temperatures. Second, they provide picotesla level sensitivity, and they have the potential to resolve single neuronal cells. So going into further details of the inverse problem, the inverse problem for the simplified cylindrical axon model is well understood. It is analytically known, but for complex pyramidal neuronal cells, it cannot be analytically solved. And the other challenge that we have is that for Fourier-based 2D reconstruction methods, the reconstruction is non-unique until it is additionally constrained with a priori information. Now, considering the diamond nitrogen vacancy based sensing current capabilities, the diamond NV based sensors, these are exactly magnetic field analogs of light microscopes, as in they can detect microscale magnetic field patterns with sensitivities of per pixel few to tens of microtesla and an image acquisition time of around several minutes to few hours. Which we note here is that means that only static magnetic field imaging is possible with most current state-of-the-art diamond NV centers. Recently, these diamond NV wide field magnetometers have been employed to study a number of different kinds of phenomena like magnetotactic bacteria and magnetic nanoparticle imaging in living cells, and also superconducting vortices. So before I go into the exact objectives, I'll briefly go over the diamond NV center physics. So this the diamond NV center is essentially a nitrogen substitution in the carbon lattice with an adjacent vacancy and an overall additional negative charge. So there are two unpaired electrons in this system. The ground state is a spin triplet with the sublevels ms0 and ms plus minus ones separated by a microwave energy gap. The NV center is red fluorescent. So when excited with 532 nanometer, it emits red fluorescence. And the other property is that it exhibits spin-dependent fluorescence. So if the electron populations populate the plus minus one state, the photoluminescence is much lower as compared to when the population populates the ms equals to 0 state. So in presence of external magnetic field, the degenerate ms plus minus one states are split by Zeeman effect, and these and the amount of splitting can be determined by monitoring the photoluminescence versus the applied microwave resonant frequencies. So by monitoring the dip in the PL contrast, we can exactly pinpoint what are the resonant microwave frequencies, which with an effective transduction ratio or the gyromagnetic ratio can be converted to magnetic field numbers. So this is how the magnetometry is performed with NV centers, and because of the narrow line width of these NV centers, one can perform ultra-sensitive magnetometry. So the two major objectives of this work is first to computationally analyze the spatiotemporal resolution that is offered by the proposed technique. And the second is to develop an algorithmic method that if these experimental magnetic fields are available, how do we reconstruct the three-dimensional neuronal activity? And the third is to develop an improved dynamic two-dimensional diamond NV magnetometry setup. So in the first part of the work, we simulated the two-dimensional magnetic field maps that will be measured by NV centers from pyramidal cortical mammalian neurons. And we found that the action Heller component in these mammalian neurons has a very dominant magnetic field structure, which is almost 2 orders higher than the magnetic field signatures of other compartments of the mammalian neuron. So this we found that significantly simplifies the inverse problem. And using this as a feature, we developed a matching pursuit algorithm that can iteratively take in the experimental magnetic field maps and reconstruct the three-dimensional neuronal magnetometry. We found so with this proposed algorithm, we found approximately 70% reconstruction accuracy for both neurons distributed in two-dimensional layer and in three-dimensional volume. neurons as close as 10 microns spatially close, and with spike timing differences of 1 millisecond or more were resolved with this particular with the proposed algorithm. So once we know that we have a certain algorithm that can reconstruct the three-dimensional neuronal activity, we proceed with building the quantum diamond microscope. So here is a schematic of the experimental setup that we have built. We have a diamond crystal in which nitrogen vacancy centers are present. This crystal is excited with green 532 nanometer excitation, and the emitted fluorescence is collected via an objective and imaged onto a single photodiode or onto a CCD camera, which provides us two modes of operation of this setup. One is the single photodiode mode, which acts in like a bulk magnetometer, because all the light is summed from all NV centers is summed onto a single point. And the second mode, in which this light is divided into is projected onto the CCD array, and it gives us the microscale pattern. In both of these, we use lock-in detection, and specifically over here we use a lock-in camera where each pixel performs lock-in detection. Microwave excitation to these NV centers are provided through microwave antenna, and calibrated magnetic fields are provided through multi-turn coils. Both the optical and the microwave excitation in this setup are gated to hundreds of nanoseconds using optical and microwave switches. And this entire experiment is controlled by a high-speed pulse generator, which precisely times and controls all the excitation pulses and the data acquisition. We developed custom softwares in MATLAB to design and perform these experiments. Now coming to the main part, wherein we want to develop a setup which is dynamic, and therefore we need significantly higher significantly higher SNR in our diamond NV magnetometry experiments. And therefore, we use lock-in detection methods. So here, as you can see, we have an NV resonance curve, and by modulating the microwave excitation frequency in an oscillatory manner, we can create NV photoluminescence in a very narrow band, localized in a very narrow band frequency, which can be further detected with a lock-in with the lock-in detection technique. And we can see that a typical experimental noise spectrum looks like this, wherein most of the noise in the experiment is concentrated in the DC or the 1/f regime. So using lock-in detection, we can actually take our sensing regime to a higher frequency where the noise level of the setup is much lower, and that allows us to do fast temporal magnetometry. So here we see the wide field optically detected magnetic resonance features. So all two-dimensional pixels have been concatenated into one dimension here, and we can see that multiple pixels have resonant frequencies. The 8 resonant features are coming from NV centers that are oriented about the 4 different tetrahedral axes. So each NV center splits into 2 peaks, and we get 8 peaks here. So here are some example pixels which show these resonant features. Here we show that depending on the camera acquisition parameters and how fast we modulate, which also governs the imaging frame rate of our setup, the shape of the NV resonance might change. So with this improvement, mainly by the virtue of the lock-in detection that we use, we were able to perform dynamic magnetometry. So first, we see in the bulk magnetometry mode. So one key difference from the static acquisition to the dynamic acquisition is that in dynamic magnetometry mode, we fix one single microwave frequency to the resonance point, and any external magnetic field fluctuation shifts the resonance, and that can be scaled with a factor, and the fluctuations in the voltage directly yields us the magnetic field fluctuations. So here we can see tracking of a 25 nanotesla field in the single point mode. Now the same thing can be extended to multiple wide field pixels. So here you can see that for a 33 microtesla field, we have wide field imaging frames that are arranged, and and a square magnetic field pattern was fluctuation pattern was applied, and we see that the multiple pixels are tracking this magnetic field pattern. So this to highlight is one of the key improvements that we have been able to achieve is to be able to perform millisecond scale dynamic magnetometry, in contrast with one of, for example, an example study wherein we see that imaging of these nanoparticles are of several minutes time scale using conventional CCD cameras. Now, but this to further improvise the claim, what we also need to show in this dynamic magnetometry is to be able to resolve some microscale features in our microscope. So with that, we install some main upgrades to the setup. First, in order to plug in, initially all the data that was taken was on a horizontal setup, which is shown here around July 21. So after this, in order to incorporate microscale samples, magnetic field samples into our setup, we developed a vertical wide field microscope configuration, which is shown here, wherein the square part highlights the wide field microscope part that we have imaged. And another key part that we have here is that initially our sample had nitrogen vacancy centers distributed all throughout the bulk volume of the crystal. So that is replaced by a crystal wherein the NV centers are concentrated in 1 micron layer. As a result of that, the magnetic fields from the sample is not averaged over the thickness of the diamond sample, but is averaged over only the thin NV layer thickness, which is just 1 micron. As a result of that, that improves the spatial resolution of the magnetic field images. Some of the other sample mounting considerations are, so to show an example sample, so here is an image of a micro scale of a gold microcoil that is fabricated by Anuj. So we have this small microcoil through which micro scale current will flow. This sample is sort of inverted and brought into close contact with this 1 micron high density NV layer to minimize the standoff. Minimum standoff allows maximum resolution of these magnetic field features. With these, so when there is a microscale current flow on a very small micron level, there are heterogeneities produced in the resonant frequencies of different nitrogen vacancy pixels. These resonant frequencies are then further converted to magnetic field images, and through a very old established method developed by Roth et al. For squid magnetometers, these the magnetic field information can be converted to current flow density information. So here we have a 10 micron micro wire, gold micro wire with a 90-degree bend, and we can see that we are able to image the current flow in this micro wire. So here are certain specifications of the wide field microscope that we have developed. The field of view is 150 microns cross 150 microns. And the per pixel resolution here is 1.33 microns, which can be improved by changing the imaging objective. And the sensitivity that we achieve is approximately 3 microtesla per pixel. The scale bar here is 13 micron. So to move towards conclusion, with this capability, the microscope that we have developed, we intend to image these spiral gold microcoil structures, which essentially, because of the concentration of the magnetic field in this region, they will mimic the action Hillock magnetic field signatures that we found, as I initially discussed. Then further, with multiple stacks of these spirals, we intend to perform volumetric magnetic field imaging and test the algorithm that we have developed in our earlier paper. Some of the other extensions are that in our current setup, we can do some fundamental electron spin ensemble experiments. For example, I have shown some Rabi oscillations and Ramsey oscillations, that have been obtained on our setup with some coherence time, with T2 star coherence time of around 800 microseconds. and short noise sensitivity of 5 for the microtester. We can see that these Ramsey fringes shift with applied external fields. So this is a summary of the output over the last 5 years of my PMRF tenure, and these are the acknowledgements. And I'm open to questions.
Thank you, Madhur, for nice presentation. And now this presentation open for question. Anybody in the audience side? Madhur, well presented, and I have a few basic questions. And so now you are talking about the magnetic properties, the neurons. You can image, right? Can you image the other than neurons without the magnetic? Can you do that?
So in this microscope, because we can probe, it is sensitive only to magnetic fields. We can only probe magnetic field patterns that are generated by the neurons.
Yeah, okay.
Other samples with magnetic fields can also be probed.
Other samples can be used, right?
Yeah. For example, I showed an example image of superconducting vortices have been probed. And in geological samples, there are certain remnant magnetizations that have been probed by people on the same diamond nitrogen vacancy platform. So these are the two examples. Magnetic nanoparticles in living cells have also been probed.
That should have the property of magnetic field property should be there. Magnetic property the cell should have, or it's a prerequisite the magnetic property, or any cell can be used. The cell should have getting the point?
Yes, the cell should have some particle that should be magnetic to essentially be captured on this microscope.
Can we do it on the in vivo or in vitro system? Can we use it live streaming kind of thing?
Yes, for in vivo cells, there has been demonstration of this setup. In fact, the example that I showed in the presentation was performed in living cells.
Living cells, live cells. Oh, very good. Very good.
The only consideration is that we need to take some heating considerations because to gain the sensitivity in the setup, we generally use high optical powers. So when we probe some living cells, we need to do additional cooling of the diamond crystal.
Right. Okay. I was indicated there only one question, quick question, because the time is up. What is the role of green laser? What happens if we use slightly lower wavelength laser than wavelength laser, say for 464 nanometer blue laser?
So NV centers have been shown to be excited on other wavelengths as well. So the best excitation, the resonant excitation, is at 637 nanometer itself. At lower laser excitation, the fluorescence will reduce.
Okay. Hope it is the question is answered. Yeah, because of that Agnes is telling time is up. So with that wonderful presentation, we'll come to an end of this presentation now. I would like to thank all the presenters. They have stick to their timing, from Darshan, and then Himanshu, then Palomi, and then Kandesh Agarwal, and finally Madhur. So they have all given a good presentation, wonderful presentation. Thank you all for giving a wonderful presentation in this symposium. And I would like to thank the organizer for the opportunity to listen to them and share the session. Thank you, audience, for participating in this and you are contributing towards the questions and answers. Thank you so much. And with that, we come to an end this program, this session. I hand over to the organizer. Thank you. The organizer, any organizers are there?
Okay, it's fine. Thank you, sir.
We are beginning the next session. So can I start?
Yes, ma'am. Please start.
Yeah, muted.
Yeah. Thank you, Professor Rajamani. So we're beginning the next session. Thank you so much for
Yeah,
conducting the previous one.
Thank you.
So in this session, we have 3 presentations, and most of them, are related to some biomedical applications. And the first speaker in this session is Saswat Chaudhary, who's a PhD student at Biosystem Science and Engineering, IC Bangalore. And Saswat is going to tell us about his work on 3D printing of polymers for biomedical applications, engineering biliary stents. So welcome, Saswat. So you have 15 minutes, Saswat. So try to finish in that 15 minutes. And any question and answers from the audience, please try to type it in the QA and box so that we can read it out at the end of this talk. Thank you. You can start.
Thank you, Professor Singh. I'll keep it within the time limit. Can you confirm if my slides are visible and I'm audible?
Yes, sir. Please make it.
Yes. Perfect.
Okay. So very good evening to one and all. Thanks for this opportunity. So my talk will be titled as 3D 4D printing of absorbable and deployable medical devices. And I work with Professor Kaushik Chatterjee and Dr. Sonal Asthana at ISC Bangalore. Giving a brief historical perspective of medical devices in general. Coming to the medical devices, there have been huge innovations in the medical devices in the last century, and we have seen resorbable as well as deployable medical devices coming to the forefront. Here is a typical bone fixation device made out of PLLA, which is completely resorbable and absorbed in the body in a finite period of time, nearly 1 to 2 years. And this is yet another medical device made out of PLLA hydroxyapatite composite, which is again used in bone fixation devices. Now, these are specific class of medical devices which are called absorbable, and they have the advantage that the patient doesn't require a second surgery to remove the implant once the function is served. Now, the highlight of a deployable medical device is that it can be deployed in the body or put inside the body in a minimally invasive manner. For example, this nitinol stent, which is made by Phenox company, and it is used as an intracranial stent. And of course, there are biliary stents and vascular stents as well, which are deployable, but they are not absorbable as of now. There are very few examples of materials which are both absorbable and deployable. So this is one such example of, I hope you're able to see this part. Okay, so this is again about vascular stent made of PLLA, which is both absorbable and balloon deployable. But as you can imagine, there are not many such examples of devices which combine both these highlights of medical devices, which would be a surgeon's dream. And we are trying to address some of these clinical challenges. Okay, so the broad objectives of our project is the following. So if we consider medical device to be the central highlight, the first example is, the first highlight of our research is to make it absorbable, which will prevent a second surgery to remove the implant. It can completely absorb in the body by its own. Secondly, it should have the right and optimum mechanical properties with sufficient strength as well as flexibility to match the mechanical properties of the native tissue. And coming to the manufacturing side of things, we want to fabricate the medical device by 3D printing so that we can get customized patient-specific shapes and sizes. And lastly, we also plan to make the material to be of shape memory, which means that it can be fixed into a temporary shape. And upon triggering with the right kind of stimulus, it can be recovered to the initial shape. And it helps us to implant the medical device in a minimally invasive manner. Now, this shape memory plus 3D printing combinedly can be called as what is known as 4D printing. So coming to the first highlight, which is absorbable polymers, we chose polyesters class of polymers, which are mostly polylactic acid, polydioxanone, and polycaprolactone, because they are biocompatible and bioresorbable, and are FDA approved for use in large scale production of various medical devices. So this is the extrusion-based 3D printing we have in our lab, and we use this to prepare different kinds of patient-specific shapes using some of those polymers. So now coming to the last highlight, which is shape memory mechanism in polymers, just to give a brief background of this. So a polymer is called to be shape memory if it has basically two components in its molecular architecture, which is net points and switching segments. So this is the original structure of shape memory polymer. It can be programmed by heating it above its transition temperature, which is usually the glass transition, or it can be melting temperature as well. Now, this is fixed at a higher temperature, which is called temporary shape. Now, once we cool it back, it is able to fix this temporary shape even at a lower temperature. And then once we give a thermal stimulus above its glass transition, it is able to recover back to the original shape. So this has this entire process is entropically driven. And as you can see, these two components of molecular architecture, they contribute towards this shape memory. So we chose PLA as the first candidate for our applications because it is both absorbable and it can be, it is also shape memory. So this is the original as printed shape you can see here. This is programmed and deformed into a compressed shape like this deformed shape. And upon recovery at around 70 degrees Celsius, it is able to go back to its original shape to some extent. And here is the video of the same. This is the deformation at higher temperature, and this is able to stay at that. It is able to fix the deformed shape at room temperature. So once we heat it back, as you can see, this is able to deform back to the or recover back to the original shape. So, but the disadvantage of PLA being a shape memory polymer is that it only shows minimal shape recovery of around 80%, and it also requires a considerably high temperature of around 70 degrees Celsius, which is not feasible for any biomedical applications because it will simply burn the tissues. So, which is why we shifted to a different kind of polymer, which is a copolymer, and it's named as PLA-MC. This is the chemical structure. It has a lactide repeating unit and a trimethylene carbonate repeating unit, and together it forms a copolymer. And these are various 3D printed structures we could obtain by 3D printing. And this is a typical thermal characterization of PLMC. As you can see, this has a melting point around 150 to 170, and this has a glass transition between 15 to 25 degrees Celsius from this inflection point you can see. Based on that background, we again fabricated different kinds of shapes to start off, simple 2D shapes. As you can see, this can be nicely deformed and then recovered back into the original shape. And we chose different kinds of shapes for that matter, just to prove the proof of concept. And this is one typical video of the star shape. As you can see, the deformed shape was able to recover at very low temperature of 40 degrees Celsius into the original structure. I'll just play it again. So this was the deformed shape, and it is trying to recover to the original shape in a matter of seconds, few seconds. So this was the PLA-MC, which could be both absorbable and deployable because of its shape memory property. And it shows considerably high amount of shape recovery, around 99%. And the time frame of recovery is also very short, around 15 to 20 seconds. Now, this brings us to the application side of things, that how can we employ this property of shape memory into making a medical device. So to start off, we fabricated different kinds of 3D scaffolds using this PLA-MC of different infill patterns and porous pore sizes. And as you can see, this is the original as printed disc kind of scaffold, which is not deployable in its initial configuration. As you can see, it is not able to be fitted into this tube. But once we deform it slightly above room temperature, this deformed disc can be easily deployed into this tube. And upon thermal recovery at around 40 degrees Celsius again, this is able to recover to the original shape. So this is a very crude way of demonstrating the applicability of these shape memory polymers into tissue scaffolds. Now we wanted to make this shape recovery essentially contactless, and for that we chose Fe3O4 nanoparticles, which have a long history of being used for different medical applications because of their favorable biocompatibility and very magnetic at room temperature and various other useful properties. And they have been used for various applications, including hypothermia treatment, immunoassays, etc. But we wanted to reinforce this Fe3O4 nanoparticle into our polymer and make these composites, which you can see, because Fe3O4 particles have the capability of being inductively heated, which means they can convert the magnetic field into thermal energy. And using this principle, the entire composite of the shape memory polymer with this nanoparticle can be deployed in a remote contactless manner. As you can see from this video, this is the deformed star kind of shape, and as a function of time, this is able to go back to the original shape because of the presence of Fe3O4 nanoparticles inside this composite, which is allowing it to be inductively heated. And this is again happening at very physiologically relevant temperatures and short time. Now, similarly, we wanted to use this for tissue engineering application. So we fabricated a porous scaffold of this kind of a scaffold, which is having this original dimensions, and it can be then deformed into this compressed shape, which by the application of alternating magnetic field, as you can see from this video, this deformed shape is nicely able to go back to the original shape in a matter of a few seconds to minutes. So these pores are kind of expanding as a function of time under this alternating magnetic field, and the temperature is very close to the physiological temperature, around 40 degrees Celsius, and the time required typically takes around 40 seconds. So finally, we have done some basic cytocompatibility studies on the nanoparticles, the Fe3O4 nanoparticles. As you can see from these images, live-read images, the cells are nicely, the cells are healthy, and they also are viable for a period of 24 to 48 hours. And we plan to do these cytocompatibility assays on the polymers and composites also. So these are some of the future works. We are planning to do some mechanical properties of the composites, both static and dynamic. And we also plan to explore other stimuli for shape change, for example, light and pH. Lastly, thank you for your kind attention. And I would like to acknowledge my advisors, Tata Steel Private Limited for the funding, PMRF, of course, and the entire biomaterials laboratory. Thank you very much.
Thank you, Saswat, for keeping it under 15 minutes. So let's see if there are any questions. I have a couple of questions for you. Looks like there are no questions. Maybe people will start typing now. So Saswat, one question I have is this PLA MC that you're talking about. Have you varied what is the molecular weight for this?
Ma'am, so we haven't characterized the molecular weight exactly. So I don't have an idea on that.
But maybe you want to do that because that
Sure,
will be a very important property, right? And especially because you could use the molecular weight to vary the properties as well. So maybe you want to try if there are what is the molecular weight of this particular thing that you used, and how difference in molecular weight will affect the properties, right?
Right.
So the next question I have is how many cycles of this temperature lowering and that you can do before you lose the shape memory effect? How many cycles can you do this?
So we have not experimentally measured it using a DMA, but when I used to conduct this normal shape memory characterization tests in a heating setting, it usually goes up to 3 to 4 cycles before it loses the memory
ability. Okay. And in your previous slide with cytotoxicity, I noticed that the ferric oxide had a little bit more toxicity, or was it just a random observation? I think you have cobalt and the other one was iron oxide,
so that
right?
we had explored as another alternative nanofiller, but that didn't work out very well in terms of shape memory. So anyways, we don't plan to use it. So.
Right, but your iron oxide has some toxicity, right? There was some dead cells. There were some dead cells in your iron oxide.
Sure. I mean, compared to the other nanofiller, it has. Yeah.
Okay.
Right. But yeah. As such,
so have you quantified? Have you quantified that? Have you done
no,
any?
Not yet, ma'am. So sorry.
Yeah, so quantification, maybe you want to do quantification for that as well.
Yeah,
right?
I think the best way would be to test the polymers and composites directly with the cells instead of just the nanoparticles. So we plan to do that, and those studies are ongoing.
Okay. So there is one question in the Q&A box, which is, since the temperature at which shape changes occur is 40 degrees, which is close to the body internal temperature, what is the requirement for iron? Okay. So what is the requirement for your Fe3O4?
Right. So as I mentioned, this allows us to have a contactless actuation that without any need for direct hitting, we are able to remotely trigger the shape change. So that is the advantage of FE304.
That's right. Excellent work, Saswat. Good job. And I think continue this work, and we hope that you bring something out in the market soon. And these are all practical materials. So good job with that, and very good presentation. And you also handled the question answers well. So congratulations on that. Okay. Thank we move to the next. Yeah, thank you. So we move to our next speaker. Our next speaker is just a second. So it's Sivaprasad Ji from Aerospace Engineering at IIT Madras, and he will tell us about his work on investigation of bifurcation characteristic in suddenly expanding confined jets and development of a novel fluidic mixing nozzle. So sounds like an interesting topic. Let's hear from Sivaprasad. Yeah, you have 15 minutes, Sivaprasad.
Please unmute yourself.
Unmute, yeah. Can you unmute yourself? Looks like it's not some connection issue. Or?
Yes, ma'am. Some internet issue, ma'am.
Okay. Maybe he's trying to rejoin.
Yes, ma'am. I think so.
Good. Interrupted in between, ma'am.
That's fine. Now you're audible.
Yeah, I'll share my screen.
Sure.
Can you see this slide?
We can. Yes. Yes, okay. We can.
Thank you, Professor, for the nice introduction. Good evening, everyone. Today, I'm going to briefly present the works that I have done as a part of the PhD program on investigation of bifurcation characteristics in suddenly expanding jets and the development of a novel fluidic mixing nozzle. I will take you all through my talk via these 7 sections. I'll give you a brief description about the sudden expansion geometries and the bifurcation of the flow through them. And we'll briefly give you some applications which involving the sudden expansion geometry. Then in the subsequent slide, I will present the research gap and the objectives that we have formulated out of this gap and the work that have been done to achieve these goals. Then the last 2 sections, I will present the works that need to be done in future to complete the study and the scholarly achievements in the due course of the study. So sudden expansion models are fairly geometrically simple one, but the flow phenomena that happens inside is quite complicated because of the sudden change in the area, as you can see in these 3 figures in the schematic. This smaller duct connects to a larger duct abruptly. So the flow can't attach to the wall anymore, and it gets separated from the smaller duct to the larger duct with a recirculation. These are in these 3 categories mainly based on the degree of the freedom of the separated jet. Separated jet in this case will have only degree of expansion which constricted to this plane of the screen, that is the YZ plane. It can't expand out of this plane. So this is have only a 2-dimensional degree of freedom and also will be referred as 2D sudden expansion. But in the other 2 cases, the separated jet from the smaller duct will have all 3 dimensions of degree of freedom to expand. So it is called as 3D sudden expansion with only change is the cross-sectional area. Cross-sectional area, cross-sectional, not area, cross-sectional shape. For this geometry, the cross-sectional shape is circular, which will be called as axisymmetric sudden expansion. For this case, this is rectangular, which will be referred as rectangular sudden expansion. The non-dimensional numbers involved in these kind of flows are Reynolds number, which is defined as the ratio of the inertial forces to the viscous forces, the Mach number, which is defined as the inertial forces to the elastic forces, and expansion ratio, which is the ratio between the diameter or the height of the larger duct to the smaller duct, and the aspect ratio, the length of the larger duct to the smaller duct. Even though the geometry looks is symmetric, at least in one plane, the flow will be symmetric only up to a particular Reynolds number, which is called as a critical Reynolds number. So in this schematics, this black color indicates the geometry boundaries, and the red indicates the jet boundaries. The blue indicates the recirculation zone. So below the critical Reynolds number, that is RECR, the flow will be symmetrical. But the moment the Reynolds number increases beyond that particular value, the jet attaches either to the top wall or to the bottom wall. It's also called as pitchfork bifurcation. These flow characteristics are desirable in combustion application, but in certain other application, they are undesirable and should be avoided or eliminated to the minimum possible extent. So I have listed a few of these applications, which is one is the gyrotherm burner. Gyrotherm burner is patented by the FCT combustion company, which uses the bifurcation to enhance the mixing. Whereas in high altitude testing facility, we have one in our ISRO, and this is the picture from DLR Germany. The rocket nozzle has to be tested before actual firing. So we need to test it under space-like condition. For that, we need low pressures. For that, in high altitude testing facility, what industry people are doing is it will be kept inside a larger chamber and will be fired. So with the jet momentum itself, it entrains all the fluid. Since the momentum is so high into the supersonic regime, the flow will be, the pressure near the nozzle will be vacuum, and which eliminates a requirement of a vacuum pump. In combustion chamber of a gas turbine engine is a sudden expansion. The flow from a compressor, flow from the compressor has to be decelerated to prevent the flame blowout. In aortic stenosis, in heart also, the same sudden expansion flow is happening. This is in biological systems. So what happens is that aortic stenosis is a condition in which aorta will be neither fully closed nor fully open during its open and closed states. But in a healthy person, when it is closed, it should be closed fully, and it should be open fully when it is in an open state. But so the blood flow behaves like a sudden expansion. So after going through all the literature, we have found that the reason behind the 2D and 3D sudden expansion, there is a characteristic change in the 2D and sudden expansion, and the reason behind it is unknown. In axisymmetric sudden expansion, the studies concerning the expansion ratio and the dependence of the expansion ratio on the bistability, that is also scarce. And a precessing jet instability is there in axisymmetric sudden expansion at higher d by d ratio, which is not there in the lower d by d ratio. That is also unexplained. How can we use this bifurcation phenomena to develop a better and efficient fluidic mixing nozzle for air breathing propulsion system? So out of these gaps, we have formulated our objective as experimental investigation of jet bifurcation, axisymmetric and planar sudden expansion geometries for varied expansion ratio and inlet flow speed, which will be measured using LDV, flow visualization, PIV, pressure measurements. Computational study of jet bifurcation in axisymmetric and planar sudden expansion using RAN simulation. Devising an efficient and cost-effective mixing enhancing device. The quantitative study of the flow field in that geometry with that mixing enhancing device. So the hypothesis for the hypothesis for the difference of the 2D and 3D, that is the axisymmetry case and the planar case. The characteristic difference in the flow characteristics, we are associating it with the recirculation bubble characteristics. As I said, after a Reynolds number critical value, it should attach asymmetrically. But that is actually happening in planar sudden expansion only. The degree of that asymmetry is very less in the axisymmetric sudden expansion. We are trying to investigate by associating it to the recirculation characteristics. By zooming into this is a 3D simulation, it's a RAND simulation result, and we can see that this blue line indicates a streamline and red line indicates the vortex course. So here in the zoomed in version of this axisymmetric sudden expansion, the recirculation zone is actually connected with another recirculation zone, which is a sole vortex. But you can see that in a planar sudden expansion, that two recirculation zone is distinct by the presence of the central jet. So this makes the recirculation bubble to grow independently. But whereas here the recirculation bubble allows the jet to grow uniformly. So to test this hypothesis, we have fabricated a axisymmetric sudden expansion and attached the plates in the recirculation zone to detach that communication. For that, and to prove this, after this, what we have done is we have taken the velocity profiles at different axial locations. This axial locations is from the expansion corner for two different Reynolds number. This is measured using laser Doppler velocimetry, LDV. So this is a velocity profile. In the y-axis indicates the velocity, axial velocity, and x-axis indicates the axial location. And see that without the plate, it is symmetric, as I explained. But by the attachment of the plate, in all the cases, the maximum velocity now tended towards the walls. So this proves the hypothesis that the connected recirculation bubble is the reason for the suppressed asymmetry in axisymmetric sudden expansion. What about the planar sudden expansion then? If the hypothesis is true, then in the case of planar sudden expansion, if we connect the recirculation bubble together, then we should be able to achieve a symmetric flow in that geometry also. So for that, what we have done is we have fabricated a planar sudden expansion with an expansion ratio of 2 and attached a barrier plate to allow this communication to happen. And the TS represents the thickness of this plate. Okay, so the same procedure is repeated for two different Reynolds number and two different axial location. This axial location A represents x equal to 200 and B represents x equal to 305 for both the Reynolds number. You can see that without a plate, it is a planar sudden expansion where the jet is actually near to the wall. But with the introduction of the plate, the jet became symmetrical. As you can see in all of these cases, the jet became symmetrical. That proves our hypothesis for both the cases. So then we have here the TS, the 10 mm is taken arbitrarily. We checked that thickness also, whether it matters. So we have found out there is a critical thickness which will only be able to stabilize the flow. The thickness is found out to be 5 mm for this particular case, and the work need to be continued further. And based on this proven hypothesis, we have come up with a flow mixing device, which will enhance the mixing, large-scale mixing of the fluid. So it is called SEM, that is abbreviated like this, and it is flow switching suddenly expanding mixing nozzle. As you can see, it's a planar sudden expansion with a movable barrier plate. So it will be rotated using a motor, and its speed will be controlled using a PWM, and which will be monitored using a Hall effect sensor, which will be further connected to the Arduino microcontroller. Here in the bottom video, you can see that the SEM operation in action. This is when the motor rotates at 148 RPM. This video is slowed down for the presentation purpose. You can see that when the plate moves down, you can see the jet is actually attaching to the bottom. But when it moves up, it becomes symmetric. It comes to the center, as you can see here. Wait for it, it will become symmetric. the jet is becoming symmetric. So so it came out of the wall and became symmetric. This enhances the mixing. So how much enhancing it can impart to the mixing. So for that we have to measure the velocity profile and we have to calculate the jet spread. That's been done in different axial location at the exit of the nozzle, and spread, that is the jet width, is plotted along that axial location. We can see that the flapping case is far better than the non-flapping case for this particular plate width. Then plate width is changed to 10 mm, and we have found the same trend, and we have found that the jet growth rate, which is a slope of the spread, is actually far better than the 5 mm case in this, and exactly it will be 80% better than the 2D non-flapping jet, and 150% better than the 3D non-flapping jet. And it has been extended to the 15 mm plate, and we have found that the same trend exists, but the jet growth rate seems to be higher in the 10 mm case, so we have optimized for 10 mm. In the shock transition in confined jet, this is a unique observation that we have found here. When in the planar sudden expansion, when we are increasing the pressure such that pushing the flow to a supersonic extreme, the jet which was asymmetrical attaches symmetrical with an MRRR transition, which is actually at a constant angle, incident constant angle. So such transition is unique because in open jets and all, that transition happens with a varying angle. So we have linked this transition to a pressure matching condition. If that is true, then it should exist in a wind tunnel. So we have fabricated a wind tunnel with a transition Mach number of 1.5 and studied the transients, starting transients. We have found out similar transition is existing from MR to an RR transition is happening in the wind tunnel also, which proves that this is a pressure matching condition. So coming to the conclusion, we have that the hypothesis we have proposed for the suppressed asymmetry in sudden expansion compared to the planar sudden expansion tested experimentally and proved, and also found that a critical value of plate thickness is found for the stabilizing the asymmetric jet. Based on the hypothesis, SM nozzle has been invented, and the same nozzle is also optimized for the plate thickness. And the unique constant angle MR to RR shock transition during the symmetrical reattachment of this jet is linked to a supersonic wind tunnel pressure matching situation. So study involving we need to continue this study to get a complete picture of the flow field, which requires study involving the expansion ratio dependence on the bifurcation of the axisymmetric sudden expansion, the plate width dependence on the symmetry breaking bifurcation, PIV experiments to quantify the recirculation bubble characteristics in sudden expansion geometry. And these are the scholarly achievements, and thank you.
Thank you, Shivrasad, for under 15 minutes, and good presentation, because I'm not from the field, but I was able to understand a little bit of your presentation. So you made it very nicely, you related with a lot of practical applications, and you actually made it understand what your work is, which is very nice. So let's see if there are any questions. Otherwise, I'll ask a very naive question. I don't understand this, fluid dynamics that much. But you showed an example of that. This is similar to what one sees in a heart or something like that, right? I think you were trying to correlate, right? So the jet that you're talking about right now, if you have a liquid flow, then that will also depend on the viscosity of the liquid, right? Or it doesn't matter with this. Okay.
Exactly. So yeah, it depends. That's why
so go ahead. Yeah.
That's why this flow is actually characterized with the Reynolds number. Reynolds number is a ratio of this inertial forces to viscous forces. Viscous forces is nothing but this viscosity is what taken care inside.
Right. So whatever whatever you have shown with your work, can it be applied to biological systems as well, or
that's where there is one more regime that I'm working on a Newtonian flows. So blood can become non-Newtonian.
Right. That's what I was actually looking at the blood. So that will be a different study, is it? Or is it you can extrapolate things that
Actually,
you've found?
maybe we will be able to extrapolate, because I have seen many papers in non-Newtonian fluids also. The same trends exist, even though the exact values are not that, but the trends remains the same. So these features should be existing when in the case of aortic stenosis also. So this kind of blood flow recirculating inside this aorta itself is
I think you can do so many other things. Blood flow is one example. There could be flow of nanoparticles. Many people are using nanoparticles for drug delivery and other applications. So how are these colloidal particles moving, and what kind of flow is happening there with respect to their blood? I don't know. I'm just being a very naive, just thinking through.
So of course, ma'am, we can extend this as a broad application range. We can't actually limit to one particular place. Only problem is measurement varies according to each regimes. When it becomes non-Newtonian, we have to take care the viscosity changes with viscosity changes, and when the Newtonian, it will be different. So the computations that we are actually employing, the assumptions we will be putting, it's a Newtonian. So like a tau is directly proportional to the velocity dou u by dou y. So that proportionality is actually failing in a non-Newtonian fluid. So we can't actually exactly, we can't say that. So each regime, as Professor indicated, we can actually do it, but each will be another set of study. Like
Right.
Simply take it as a section and do it. It's very difficult.
It's not.
It's very difficult.
But it's interesting. That will give you some basis for those work, right? So thank you, Shiv Prasad, for this wonderful presentation. And now,
thank you, ma'am.
I invite Amay, Nitin. There are no questions, I think. I checked Q&A box. There were no questions. So thank you, Shiv Prasad. Okay, so now I invite Amay Nitin Agarkar, from Interdisciplinary Center for Energy Research at IC Bangalore. And Amay is going to tell us about his work on elucidation of fluid dynamics of bacterial movements in natural body fluids and its relation to pathogenesis. So Amay, you have 15 minutes.
Sure. Yeah. Good evening to the wide audience present here, listening to us. So I am Amay Nitin Agarkar. I completed my bachelor's in mechanical engineering from NIT Surat, and later I joined as a direct PhD student in a prestigious institute of our nation, that is Indian Institute of Science. And my research advisors are Professor Saptarishi Basu and from Department of Mechanical Engineering, and Professor Deepshikha Chakravarti from Department of Microbiology and Cell Biology. And I work in the area of fluid dynamics and pathogenesis, and which is a very little drop in the vast ocean of interdisciplinary sciences and engineering. So the title of my talk as well as thesis will be Elucidation of Fluid Dynamics of Bacterial Movements in Natural Body Fluids and its Relation to Pathogenesis. So the research lacunae for the whole series of my work is that the understanding of mechanistic way of how the pathogens transfer from one host to another is acutely limited, that we are experiencing in this COVID-19 pandemic. So the fluid dynamics of bacteria or virus laden droplet, as droplet is a very integral part of any kind of flow or aerosol. So it will be studied to ascertain the effects of accumulation, coagulation, and spatiotemporal evolution of bacteria. So it's coupling with the physiological medium like blood, mucin, and all other fluids will be studied. And we will try to answer it, that why it is enhancing infection or how it is surviving in such environmental conditions. And these biological fluids can also change the gene expression in them, leading to enhanced infection. So our today's discussion will be on, this is our first line of work, and this has been published in Nature Communications Biology. So our discussion will be limited to fomite-mediated pathogenesis, that is augmented virulence of flow governed by Salmonella typhimurium. So as you know, that droplet is an integral part of any aerosol or any flow. So these droplets settled on the fomites are considered as most significant modes of microbial transmission. So Salmonella typhimurium has been chosen here as it is the most virulent pathogen. And in the countries such as Asian and African countries, nearly in 1 year, 77,500 deaths are reported. So it is that much virulent, and it causes a foodborne disease, that is gastrointestinal disease in humans. So the ability of Salmonella typhimurium to survive on inanimate surfaces such as fomites at room temperature enhances its risk of consumption. So the one sentence summary of the whole work that I will present is that the drying of bacteria-laden droplet on fomite evolve unique deposition patterns enhancing pathogenesis due to the stress experienced by the bacteria. So this is the overall schematic of our whole study. So here what happens is a fecal transmission by an animal which is infected by Salmonella typhimurium. It goes down on the ground or somewhere as a fomite, and then due to in contact with some irrigation pipeline or somewhere, aerosols are formed. Such aerosols transfer and settle on some surfaces containing bacteria, which are very much dangerous for human contact or any other animal contact, may be consumed by animals. And later on, the whole cycle of this infection goes on and on. So we tried to complete a story and form a story. So what we did is we took 4 kind of solutions, that is base solutions, which contained the bacteria Salmonella typhimurium having 2 strains, that is its wild type containing flagella, and delta flyci knockout, which does not contain a flagella, and also the dead bacteria were used. So the 4 fluids which were used are saline, milky water, dextrose, as well as mucin. So these all 4 fluids are physiologically significant as they represent some gastrointestinal fluid, IV fluid, as well as the respiratory fluid. So what we did is, using a micropipette, we just drop casted the drop on a glass slide, and we observed its evaporation, that how it evaporates, the flow velocities of the evaporation, and the fluid dynamics part was studied. It takes nearly 3 to 7 minutes for all the fluids to evaporate. Later on the dried precipitates, we took the confocal image as well as the SEM image of that. And then that dry precipitate, after some 5 hours, and we extended the study up to 72 hours, that were rehydrated, and we saw the survival of the bacteria in that. After that, we also studied its infection in the raw macrophage cell line, as well as we tried to confirm the infection study via in vivo experiments in mouse. So this is how it completes the whole cycle. Also, we studied the proliferation in door handles, that is inorganic substance, as well as organic substance that are found in fields such as tomato and cucumber. So this is the whole experimental diagram. So now we will dwell in more. So we observed the precipitates of droplet in different medium, like saline, millicue, dextrose, and mucin. So here the dead bacteria, we can say that dead bacteria, the deposition of edge is very broader, means it is spreaded full after the droplet evaporation. But for the wild type as well as delta flyci, we can say that the edge band is very narrower and dense. It is densely at the edge. So as you can see in figure number B and figure number C, the 20 micrometer is the edge width of the dead bacteria, as well as the 7 micrometer is mostly for the wild type as well as the delta flyci. While for the dextrose and mucin, we can say that the bacterial deposition is all over the droplet, all over the evaporated droplet. So we just tried to see, using the different concentration of dextrose as well as mucin, using a high as well as low concentration. So the high concentration showed a large, broader edge deposition rather than the low concentration, that you can see from the profilometry data in figure D and figure E. So by this, we can say that the solute concentration also plays a good role in the bacterial deposition patterns. So we tried to dwell more into the edge by looking at the 63x images of the edge for the saline, millicue, dextrose, and mucin. Here in the saline and millicue, we can directly observe using the naked eye, like that for dead bacteria, it is deposited all over after the evaporation, while for wild type and delta flyci, we can see very densely populated edges there. And the edge deposition is very well, and it is very narrow. So for dextrose, it is deposited broader all over because it has a soluble manganese flow that will come, I will show later in flow visualization. But in mucin, we can see that there is a polymer formation which entraps the bacteria and does not allow it to go towards the edge while it evaporates. So due to this entrapment, the bacteria is dispersed all over the droplet. So now we will study look after the flow visualization, where there is saline solution, mucin solution, and saline and mucin, where we have taken and plotted the flow vectors using micro PIV for initial time frame, that is from 0 to 50 seconds. And we can see that for saline as well as mucin solution, it is dominated by capillary flow. So most of the bacteria will be driven towards the edge in that. But for the dextrose solution, what we did is we observed a soluble melanganic flow. So we rechecked it by using it at the middle, the micro PIV, and setting it at the height of 0.7 H0 as well as 0.1 H0. So you can see in the figure number D here in the main figure in the dextrose solution. So the pink line depicts that the velocity vectors diverge from the center, as well as the blue line depicts that the velocity vector converges towards the center. So this suggests a kind of toroidal flow inside the droplet during its 0 to 50 second. Later on, we tried to see at the edge. We tried to focus on the edge also, and we saw that for very high concentration, there is a bifurcation region. So bifurcation region means what happens is the capillary flow dominates and it takes the bacteria towards the edge. But due to the soluble melanganic flow, due to the concentration of solution, what happens is there is a flow towards the center. So when this both flow meets, there is a region called as bifurcation region formed there, which has a null velocity. So here the bifurcation region comes very early in the very high concentration, that is 5% in dextrose, and it comes later in the 0.9% concentration at 0.95% of the time frame. So the results of the flow visualization were kept here, as you can see in the figure number A, where the dextrose as well as saline behaves similarly and the rush hour effect dominates at the end. So for the mucin, the rush hour effect comes very much early at high concentration because it starts forming a polymer layer and entraps more and more bacteria. So we also tried to see the contact angle variation, where by side videography of the droplet in shadowgraphy, we saw the contact line analysis of the droplet. So initial angle of contact was kept as 40 degree, and then we can see all show the similar trends, that is salt, mucin, dextrose, all the three medium showed a similar trend. But compared to without bacteria base fluid, what we can say is that the base fluid started deepening after a certain amount of time, but the fluid containing bacteria didn't deepen. That suggests us that the bacteria has a tendency to hold its edge. So that gives us a very good sense. Later on in figure C, we can see the final evaporation time, where for saline it took nearly 400 second times, and the rests are given there. So after the dried precipitate, we tried to know the survival, that how much of the bacteria did survive after this kind of evaporative stresses on a simple glass slide. So we did experiments for saline, milicu, mucin, and dextrose. So here the sample which was dried, and that is a 0.5 microliter droplet which is dried, that was again rehydrated. The rehydrated with 100 microliter of PBS, and that was plated. So here we observed in saline and milicu that both the variants, wild type and delta flyci, the bacterial count decreased from 10 to the power 5 to 10 to the power 2. So it suggests that this is a nutritionally neutral medium because it does not provide them nutrition, while mucin and dextrose will provide nutrition. So there are a nutrition rich medium. So we have also bifurcated the study in that part. So now when we dwell up to the mucin, for both the concentrations, we can see that for wild type as well as delta flyci, the survival was up to 72 hours. And this data is very much confirming to the reports that, and it shows the tactics by which the Salmonella typhimurium survives for so long periods. Later on, for the dextrose, the delta flyci variant was not at all able to survive after 72 hours. So that shows that due to the nutrition deficiency, it was not able to survive. So later on, after the viability studies, we tried to see the infection study. So what we did is we used the cell line, that is raw 264.7 cell line, and tried to give infection in that and did infection studies. So the percentage phagocytosis of post evaporation and pre-evaporation samples, you can see a very large difference is there. That means that Salmonella typhimurium, even after the evaporative and the nutritional stresses, is able to invade the cell wall of the cell lines and is able to go more inside as per the phagocytosis. And after that, on the right hand side, you can see a fold proliferation figure, where for the mucin, it suggests a very good result that after going inside, it is more able to more proliferate even after going inside. So that indicates that Salmonella typhimurium's virulence characteristics have been enhanced due to this evaporative and nutritional stresses. So later on, we tried to do the in vivo study of this using by orally gavaging the Balpsy mouse, that is as a mouse model used for Salmonella typhimurium mostly. So by orally gavaging it with the solution that is the evaporated as well as pre-evaporated, we tried to confirm that how much infection is there. So we sacrificed the mouse after the 5 days, and later on we can see that in the liver and spleen there is a considerable amount of infection, and that shows that the virulence of Salmonella typhimurium is increased due to the evaporative stresses. So to have a physiological viability of our study, what we did is we also checked the fold proliferation on the organic and inorganic substances, and here mucin 0.6 weight percentage on the right hand side is shown, is on just on a glass slide. So you can see that there is a 5-fold increase in the fold proliferation on a door, simple steel door handle, than that of the normal glass slide. So, and also for tomato and cucumber, it is 2.5 times fold proliferation increase. So that this data shows and convinces us that it can be more dangerous when it comes to real life experiences. So the conclusion derived from this overall study is that in the neutral media, there is capillary dominated flow during evaporation, and the bacteria is carried towards the edge. So dead bacteria does not form any aggregates. The wild type and delta flyci have more compact structure and are more aligned at the edge rather than the dead bacteria. And although the viability is reduced by orders, post-evaporated STMWT bacteria shows an increase in percent of phagocytosis, depicting a very higher entry percentage inside the raw 264.7 cells. So this increased bacterial virulence is governed by the flow stress rather than just the nutrition. And the potency of the fomites in causing an infection is confirmed from the observations of bacterial load in liver and spleen of the Balpsy mouse. And the hyperproliferation of bacterial burden is also detected from the commonly found surfaces such as steel door handle, surfaces of tomato, cucumber, confirming there is enhanced virulence of post-evaporated Salmonella typhimurium. So thank you very much for giving a ear to me, and I welcome your questions.
Thank you, Amay, for this very nice presentation, and very, very interesting, especially with the pandemic. I think everyone is interested in learning about what fomites are and how fomites are important in our day-to-day lives. Anyways, so let me see if there are any questions in the Q&A box. Looks like there are none. I will ask you a very simple question. So this is a beautiful study that you've done where you're showing how these fomites survive on the surfaces and what is the mechanism that they use, right, for survival and all that. But now from your study, can you come up with a solution for avoiding these fomites? Like how do you anything that we could do to avoid it based on the studies that you've done? Any solutions that you offer?
Yes, madam. Means that is in the future line of our work, that any solutions that can be done. So that can be done using some kind of what we call as giving more bacterial stresses, means by other kind of stresses that can be done.
Okay. But this should be true for any microorganism, right? I mean, what you're talking about is just simple evaporation-based, carrying of the fomites. So this should be true for any bacteria or anything, right? Any microorganism, or is it specific to?
It is specific to Salmonella typhimurium because that is a very clever bacteria in all of them.
They're able to survive. Yeah. They're able to survive.
It is very clever bacteria in all of them. So it can be done because there are escape pathogens. You may know that escape pathogens are there. So this study can be extended with that also.
Right.
And it may prove to be more dangerous in the hospital environments and all if such fomites come.
That is what I was wondering. That is what I was wondering, because this is a beautiful, I mean, this happens, right? The evaporation based method, that observation that you've done, it's a very common phenomena that I would assume that might be seen with other species also. So I think this is very interesting work. Okay, so good luck with that, and we look forward to seeing more work from you, and we'll be following it up now, looking for your papers in future.
Thank you.
So with that, I think we come to an end. There are no other question answer sessions in the QA box. And I would like to now thank all the speakers, today, starting from Saswat, Shiv Prasad, and Amer. Thank you so much for sharing your work with us and telling us about your progress. And we look forward to seeing some more good work and some impactful technologies coming out from your labs and your work. So with that, I would like to end this session. I think there is another session which probably started. I don't know how they're moving, but thank you, everyone. So shall we just end this, or how do we go about it? There is another speaker line. End the session and go to the other one?
Ma'am.
Okay. So see you all there. Okay. Bye-bye.